Right now, as you settle into stillness, something extraordinary is happening beneath your awareness. Deep within you, trillions of cells are asking a question they've asked every moment since you began. How do we keep going? Imagine your body as a living galaxy. Each cell, a tiny world with its own atmosphere, its own power stations, its own libraries of ancient instructions written in molecular code. These worlds communicate across vast inner distances, sending signals like starlight, maintaining the ordered complexity that makes you, you. Tonight, we're going to drift through that inner galaxy together, not with urgency or alarm, but with the gentle curiosity of travelers who have all the time in the world. We'll explore one of the most fundamental questions biology has ever asked. Why do living things age? This isn't a story about fear or decline. It's about understanding the river of time as it flows through matter itself. The science of aging has transformed in recent years from speculation into clarity, revealing not one single cause, but a symphony of interconnected processes, each playing its part in the passage from youth to age. You don't need to stay awake for all of this. In fact, the opposite is true. Let these words be a gentle current carrying you toward rest. The information will unfold whether you're listening consciously or drifting at the edges of sleep. Your only task tonight is to be comfortable, to breathe easily, and to let your mind wander through the microscopic cosmos within. There's no test at the end, no urgency, just the quiet pleasure of understanding a little more about the biological inheritance we all share, the slow transformation that connects every living thing to every other, across species and centuries, and the patient, indifferent beauty of natural law. So settle deeper. Adjust your pillow one last time. Let your breathing slow. And let's begin our journey through the science of aging, together, at the pace of falling into sleep. Welcome. Truly, welcome. I'm so glad you're here. You've found the Cosmic Atlas, a place where science becomes a lullaby, where the complexity of the universe unfolds at the speed of sleep. This is a space designed for the strange and wonderful overlap between curiosity and rest, where learning doesn't require alertness, where understanding can arrive in the borderlands between waking and dreaming. Before we drift too far into the night, I want to ask something small of you. If you've found comfort here before, or if something in these first few moments feels right to you, consider leaving a quiet mark of your presence, a like, a subscription, a share with someone who might also find peace in falling asleep to science. These small gestures help this work find others like you, people who want to understand the world but also need permission to rest while learning. I don't say this to interrupt the con we're building, but because this space exists in conversation with you. Your engagement, gentle as it is, tells the algorithms that there's value in slowness, in depth, in treating complex topics with both rigor and tenderness. So, if it feels right, take a moment, then settle back, knowing you've helped this small corner of the internet remain a sanctuary. Tonight's topic is aging, not the surface story of grey hair and birthdays, but the deep biological narrative written in molecules and cells and time itself. We're going to explore the 12 hallmarks of aging, a framework that represents decades of research distilled into clarity. Think of them as 12 windows into the same house, 12 angles of view on the same fundamental process. This episode will be long, perhaps 2.5 hours, maybe more. It's designed to accompany your entire descent into sleep, and to be there in the deep night if you wake briefly and need a familiar voice, to guide you back under. You won't remember all of it. You're not supposed to. Some of it will sink in during attentive moments. Some will wash over you like distant music. Some you'll sleep right through. All of that is exactly right. My intention is simple: to hold space for you to rest, while simultaneously offering your mind something genuinely interesting to process at whatever level of consciousness feels natural. Science as meditation, learning as lullaby, understanding as a pathway to peace. So let's set our intention together. Tonight, you have nowhere else to be, nothing to achieve. No deadlines, no tests, no performance required. You are simply here in this moment, allowing yourself to be curious about the biological processes that unite all living things. You're learning in the gentlest possible way, by listening as you drift towards sleep. Take a slow breath in. Hold it softly. Release it completely. Feel your body settling into the surface beneath you. Notice the temperature of the air, the weight of blankets, the darkness behind your eyelids. This is your starting place. From here, we journey inward, downward, through scales of size and time, toward the cellular processes that tick away beneath every heartbeat, every breath, every moment of your existence. The science of aging is ultimately a story about change, about how order gradually gives way to entropy, about how the systems that maintain life become, over time, a little less precise, a little less coordinated, a little more prone to error. But it's not a tragedy. It's simply the way things work. And understanding it can bring a strange sort of peace, a sense of being part of something much larger than yourself, much older than your individual lifetime. We're all temporary arrangements of matter, briefly organized into the astonishing complexity of a living body. Aging is what happens as that organization slowly returns toward simplicity. It's not good or bad, it just is. And tonight, we'll understand it better. Rest now. Let your eyes stay closed. Breathe. And let's begin. When most people think of aging, they think of the visible markers, the lines that appear around eyes, the silver threading through hair, the way standing up from a chair starts to require a moment of intention. These are real, of course, but they're shadows on the wall of a much deeper cave. Surface reflections of processes happening at scales far below perception. Aging, in the biological sense, is not primarily about how you look. It's about how well your body maintains itself. It's about the accumulating gap between the damage your cells experience and their capacity to repair that damage. It's about the gradual loss of resilience, the slow drift toward states that make disease and dysfunction more likely. Scientists distinguish between two types of age. Chronological age is simple. How many years since you were born? A number that climbs steadily and applies equally to everyone. But biological age is different. It's a measure of how much your body has actually changed, how much the underlying systems have drifted from youthful function. To people both 50 years old can have dramatically different biological ages. One's body functioning like someone 40, another's like someone 60. This distinction matters because it reveals something crucial. Aging is not simply the passage of time. It's a set of processes that correlate with time but aren't directly caused by it. Time is just the frame. Aging is what happens within that frame, the accumulation of changes that make an old body different from a young one. So what defines the difference? What changes? At the most fundamental level, aging is about risk. A young body has low risk of cancer, low risk of heart disease, low risk of infection turning serious, low risk of a fall leading to a fracture that doesn't heal properly. An older body has higher risk across almost every category. Not certainty, but probability. The dice are weighted differently. Simultaneously, aging is about resilience. A young body bounces back from illness, from injury, from a night of poor sleep or a week of stress. Recovery is fast and complete. An older body recovers more slowly, less completely. The buffer between normal function and dysfunction narrows. What once would have been easily absorbed now leaves lasting marks. These two shifts, rising risk and falling resilience, are not abstract. They emerge from specific biological changes happening at the level of molecules, cells, and tissues. And here's the key insight from modern aging research. These changes are not random. They follow patterns. They cluster into identifiable categories. They interact with each other in predictable ways. For most of human history, aging was a black box. People got old, their bodies failed, and no one could say exactly why beyond vague theories about vital essence or wear and tear. But starting in the late 20th century, and accelerating dramatically in the 21st, science began to map the interior of that black box. Researchers working on different organisms, different tissues, different diseases, started finding the same underlying processes showing up again and again. DNA accumulates damage faster than it can be perfectly repaired. The protective caps on chromosomes shorten with each cell division. the patterns of which genes are turned on or off drift from their youthful settings. Proteins start to misfold and clump together. The cellular recycling systems that clear out damaged components become less efficient. The sensors that tell cells whether to grow or to maintain themselves become miscalibrated. The power plants inside cells start producing less energy and more damaging byproducts. Some cells stop dividing but don't die, instead sitting in tissues and secreting inflammatory signals. The stem cells that regenerate tissues become depleted or less active. The communication between cells becomes noisy and inflammatory. The microbial ecosystems in our gut shift toward less helpful compositions. The immune system becomes simultaneously weaker at fighting new threats and more active at creating chronic inflammation. Each of these processes was studied separately for years by different labs in different contexts, but gradually scientists realized they were all part of the same story. They were all hallmarks, characteristic features that distinguish old cells and tissues from young ones. More importantly, they were interconnected. Each one could influence the others. Damage in one system would ripple out, creating problems elsewhere, which would feed back and worsen the original issue. This realization was crystallized most famously in a landmark paper, published in the journal Cell in 2013, then updated in 2023. The paper identified what the authors called the Hallmarks of Aging, a framework for organizing all the disparate findings into a coherent picture. The original paper listed 9 hallmarks. The updated version expanded it to 12, reflecting new discoveries and refinements in understanding. This framework doesn't claim that aging has 12 separate causes. Rather, it identifies 12 observable features that reliably appear as organisms age. Features that are conserved across species, from yeast to worms to mice to humans. These hallmarks are interconnected in a web of causation. Some are more upstream, more fundamental. Others are downstream consequences. But all of them contribute to the overall phenotype of aging, the collection of changes that make an old organism recognizably different from a young one. Understanding aging through this lens transforms it from a mysterious inevitability into a phenomenon we can study, measure, and potentially intervene in. It shifts the question from why do we age, which is almost philosophical, to which specific processes drive aging, and how do they interact, which is answerable through experiment and observation. And this is not merely academic, because if aging is not one monolithic thing, but rather a collection of specific processes, then in principle those processes could be targeted, slowed, perhaps even partially reversed, at least at the cellular level. This is the frontier of Geroscience, the study of aging as a biological problem rather than an inevitable fate. But we're getting ahead of ourselves. Right now, you're not here to plan interventions or chase immortality. You're here to understand, to rest, to let your mind wander through these ideas at whatever depth feels comfortable. As you drift towards sleep, so let that be enough for now. Aging is not one thing. It's a syndrome, a collection of changes that accumulate over time, raising risk and lowering resilience. It happens at every scale, from molecules to cells to tissues to whole organisms, and it follows patterns that science can now map with increasing precision. Let your breath flow slowly, in and out. Feel the weight of your body, the stillness of the room. We're going to explore those patterns now one by one, like visiting 12 chambers in the same vast palace. Each one will reveal something new, and together they'll form a complete picture. Imagine looking up at the night sky. At first, it's just stars, thousands of points of light scattered across darkness. But if you know what to look for, patterns emerge. Constellations, shapes that ancient humans recognized and named, turning chaos into story. The 12 hallmarks of aging are like that. They're a way of organizing the overwhelming complexity of biological decline into recognizable patterns, a map that lets us navigate the territory of aging with clarity rather than confusion. These hallmarks were formalized by a group of leading aging researchers in a paper that has become one of the most cited works in all of biology. The 2013 version identified 9 hallmarks. The 2023 update expanded the framework to 12. incorporating new evidence about the microbiome, inflammation, and other processes that have come into sharper focus over the past decade. The hallmarks are not 12 independent causes of aging. They're 12 interconnected features that show up consistently as organisms grow old. Think of them as 12 instruments in an orchestra. Each plays its own part, but the music only makes sense when you hear them together. More than that, they influence each other. The tempo of one affects the timing of another. A mistake in one section cascades through the ensemble. The hallmarks can be grouped loosely by how fundamental they are. Some are primary, upstream causes. They accumulate directly with time and cellular activity. Others are antagonistic, responses that are helpful early on but become harmful when chronic. Still others are integrative, system-level consequences that emerge when the primary and antagonistic hallmarks overwhelm the body's capacity to compensate. But even this categorization is fluid. The boundaries blur. Cause and consequence loop back on each other. The system is deeply interconnected, a web rather than a hierarchy. Let me name them for you now, not with detail yet, but just so you know the territory we'll cover tonight. Let the names wash over you. Don't try to memorize. Just listen to the rhythm of the list. Genomic instability, the accumulation of damage to DNA, the instruction manual in every cell. Faster than repair systems can keep up, telomere attrition, the shortening of protective caps on the ends of chromosomes, eventually limiting a cell's ability to divide, epigenetic alterations, changes in the patterns that control which genes are turned on or off, causing cells to lose their precise identity and function, loss of proteostasis, the breakdown of systems that fold, maintain, and recycle proteins, leading to toxic accumulations of misfolded junk. Disabled macroautophagy, the weakening of the cell's recycling program, which normally clears out damaged organelles and aggregated proteins, deregulated nutrient sensing, the miscalibration of pathways that sense nutrients and decide whether to prioritize growth, or maintenance, skewing toward growth even when maintenance is needed, mitochondrial dysfunction, the decline of cellular power plants, which produce less energy and more oxidative stress as they age, cellular senescence, the accumulation of cells that have stopped dividing but refuse to die, instead spewing out inflammatory signals that poison their neighbors, stem cell exhaustion, the depletion or dysfunction of the regenerative cells that normally repair and replenish tissues, altered intercellular communication, the breakdown of signaling between cells, leading to chronic low-grade inflammation and loss of coordination, dysbiosis, the shift in the gut microbiome toward less beneficial communities of bacteria and other microbes, affecting metabolism and immunity, chronic inflammation, the persistent activation of immune signaling even without infection, creating damage throughout the body, often called inflammaging, 12 windows, 12 angles, 12 threads woven into the same tapestry. As we move through the episode tonight, we'll visit each one in turn. I'll explain what each hallmark is, what causes it, why it matters, and how it connects to the others. Some sections will be longer, because some hallmarks are more complex or have been studied more deeply. Others will be brief, because the story is simpler or the science is still emerging. You don't need to follow every detail. You don't need to remember the order. If you drift off during one section, that's perfect. If you wake during another, welcome back. The structure is here to serve your rest, not to demand your attention. What I want you to take from this overview is a sense of the whole. Aging is not mysterious decay. It's a set of specific, identifiable, interconnected processes. Each one has been studied in laboratories around the world. Each one has molecular mechanisms we can describe, and each one contributes to the overall transformation from a young, resilient body to an old, fragile one. The Hallmarks framework has become the standard way aging researchers think about their field. When scientists propose an intervention to slow aging, they ask, which hallmark does this target? When they measure whether something is working, they look for improvements in these specific processes. The framework has brought coherence to a field that was once fragmented and speculative. And for you, lying here in the dark, it offers something different. It offers understanding without fear. Aging is not a punishment or a failing. It's biology. It's the consequence of trade-offs made deep in evolutionary time, when natural selection optimized for survival and reproduction, not for indefinite lifespan. The hallmarks are not enemies. They're just processes playing out according to physical and chemical law. Understanding them won't stop your own aging, but it might change how you relate to it, might bring a sense of perspective, of being part of something larger than your individual experience. Every human who has ever lived has aged. Every cell in your body is navigating these same processes right now. You are not alone in this. You are part of the flow. So take a deep breath. Let it out slowly. Feel yourself sinking a little deeper into rest. We're going to start our journey now, moving through the hallmarks one by one, starting with the foundation, the slow accumulation of damage to the very code of life itself. Genomic instability. DNA damage. The first hallmark. Let's begin. Inside every cell in your body is a library. Not metaphorically, literally. A molecular library where information is stored in a 4-letter alphabet, written along the twisted ladder of DNA. That information encodes everything. How to build proteins, when to divide, how to respond to signals, what makes your cells and not someone else's. This library is enormous. Your genome contains about 3 billion base pairs, 3 billion letters of code. If you printed it out in a standard font, it would fill thousands of books. And this entire library exists, coiled and compacted, in a space smaller than you can see without a microscope, in the nucleus of every cell. Now imagine that this library is under constant assault, not dramatically, not all at once, but continuously, quietly from dozens of different sources. Cosmic rays from space strike a DNA strand and snap it. Ultraviolet light from the sun causes to adjacent letters to fuse. Metabolic byproducts, oxygen radicals produced by the very process of making cellular energy, oxidize a base and change its chemical identity. Errors occur during the copying process when DNA is replicated before a cell divides. A wrong letter inserted into the new copy. Environmental chemicals, from tobacco smoke or industrial pollutants or even naturally occurring compounds in food, bind to DNA and distort its structure. Every day. In every cell, tens of thousands of damaging events occur. Tens of thousands. Your genome is being damaged right now as you listen to these words. Bonds are breaking. Bases are being altered. Strands are tangling. The information in the library is being corrupted, letter by letter, page by page, constantly. And yet, most of the time, you're fine. The books remain readable. The cell continues to function. How? Because inside every cell is also a repair crew. Actually, multiple repair crews, each specialized for different types of damage. They patrol the genome constantly, scanning for errors. When they find a damaged base, they snip it out and replace it with the correct one. When they find a broken strand, they realign the ends and stitch them back together. When they find the strands stuck together, they unpick the tangle and restore the proper structure. These repair systems are astonishingly sophisticated. They work with molecular precision, recognizing abnormalities among billions of normal bases, correcting errors without disrupting the surrounding code. For most of your life, they keep up. The rate of damage approximately equals the rate of repair, maintaining the genome in a relatively stable state. But here's the problem. The repair systems are not perfect. They make mistakes. Sometimes they miss damage. Sometimes they repair it incorrectly, introducing new errors in the process of fixing old ones. And over time, across thousands and thousands and thousands of events, errors accumulate. This is genomic instability. The gradual, inevitable accumulation of DNA damage faster than it can be perfectly repaired. Not a sudden catastrophe, but a slow drift toward a more error-prone genome. Why does repair fail to keep pace? Several reasons, and they compound over time. First, repair capacity itself declines with age. The proteins that perform repair are themselves subject to damage. The cellular systems that detect damage and recruit repair machinery become less efficient. There's evidence that key repair pathways slow down in older cells. meaning damage that would have been fixed quickly in youth lingers longer in age, giving it more time to cause problems. Second, certain types of damage are inherently difficult to repair perfectly. A double-strand break, where both sides of the DNA ladder snap at the same point, is particularly dangerous. Cells have mechanisms to repair these, but they're error-prone. Sometimes the two ends are stitched back together incorrectly. Sometimes a piece is lost. Sometimes the break is repaired by grabbing a nearby chromosome and copying a chunk of its sequence, which can lead to deletions or duplications. Third, some damage accumulates in long-lived cells that don't divide, like neurons and heart muscle cells. These cells can't dilute damage by dividing and passing some of it to daughter cells. They just accumulated year after year, decade after decade. Fourth, telomeres. Those protective caps we'll discuss in the next section themselves contribute to genomic instability. As telomeres shorten, the ends of chromosomes become vulnerable. The cell's repair machinery sometimes misinterprets these exposed ends as breaks and tries to fuse two chromosomes together end to end, creating catastrophic genomic ERARANGEMENTS. The consequences of genomic instability are profound. Mutations in DNA can alter genes. If a mutation hits a gene involved in controlling cell division, it can lead to cancer. Cells that divide uncontrollably. If it hits a gene involved in metabolism, it can disrupt the cell's ability to produce energy or synthesize necessary molecules. If it hits a gene involved in protein synthesis, it can lead to misfolded proteins and proteostasis collapse, which we'll cover in another hallmark. But the relationship between DNA damage and aging is not straightforward. You might think that organisms that accumulate damage faster would age faster, and organisms that repair damage better would age more slowly. And this is sometimes true. Mice engineered to have defective DNA repair do age faster, showing premature versions of age-related decline. Humans with rare genetic syndromes that impair DNA repair, like Werner syndrome or Cockayne syndrome, experience dramatically accelerated aging, looking and functioning like people decades older than their chronological age. But here's the twist. Sometimes, more aggressive responses to DNA damage can also shorten lifespan. Some repair pathways, when overactivated, trigger cell death or senescence, removing damaged cells from the population. This is good for preventing cancer. Cancer is, after all, a disease of cells that have accumulated too many mutations and lost normal growth controls. By killing or disabling cells with significant DNA damage, the body prevents those cells from becoming cancerous. But this creates a trade-off. Too little DNA repair, and you accumulate mutations that might cause cancer or cellular dysfunction. Too much cell death or senescence in response to DNA damage, and you lose functional cells, depleting tissues and accelerating loss of regenerative capacity. This is a recurring theme in aging biology. Systems that protect you in youth have costs. When active chronically, DNA damage responses are beneficial. They prevent cancer, but they also contribute to tissue decline. Evolution optimized for survival long enough to reproduce, not for indefinite maintenance. The pathways we have are good enough for that, but they have failure modes that manifest over longer timescales. Genomic instability is thus both a cause and a consequence. It's a primary hallmark, accumulating directly from physical and chemical processes, but it also drives other hallmarks. DNA damage can alter the expression of genes involved in epigenetic regulation, in protein quality control, in mitochondrial function. A cell with an unstable genome is a cell struggling to maintain its identity and its homeostasis. And across billions of cells, across decades, this instability accumulates, not uniformly. Some tissues are more affected than others. Some cells carry heavier burdens. But the overall trend is clear. The genome becomes noisier. The fidelity of the cellular library declines. And the precision with which cells execute their functions erodes. Is there anything to be done about this? At the individual level, avoiding additional sources of DNA damage helps. Not smoking, which introduces a flood of genotoxic chemicals. Using sunscreen, which blocks UV radiation. Minimizing exposure to environmental toxins where possible. eating a diet rich in antioxidants, which can neutralize some of the reactive oxygen species that damage DNA, though the benefit here is modest and shouldn't be overstated. At the research level, scientists are exploring ways to boost DNA repair capacity, drugs that activate certain repair pathways, gene therapies that deliver extra copies of repair genes to tissues, Senolytics, which we'll discuss later, that clear out cells that have entered senescence in response to DNA damage, potentially preventing the downstream inflammatory consequences. But for tonight, the main thing to understand is this: genomic instability is not a failure of your body. It's a consequence of living in a universe with entropy. Information degrades, order becomes disorder, and life, which is an impossibly ordered arrangement of matter, fights against that tide with impressive but imperfect tools. Your cells are fighting that fight right now, preparing, copying, maintaining, doing their best, and for decades they succeed remarkably well. Aging is what happens when the balance finally, slowly, tips toward damage outpacing repair. Let that sink in. No judgment, no fear, just recognition of the process. Breathe deeply. Let your muscles soften. We're moving to the next hallmark now, another aspect of genomic maintenance, this one involving the very ends of your chromosomes. Telomeres, the second hallmark. Let's continue. If DNA is a library, then chromosomes are the shelves, distinct packages into which the genome is organized. Humans have 46 chromosomes, 23 pairs, each one a long, thread-like molecule of DNA wrapped around proteins. And at the very ends of each chromosome, like the plastic tips on shoelaces, are telomeres. Telomeres are repetitive sequences of DNA. In humans, the same 6-letter sequence, TTAGGG, repeated over and over thousands of times. They don't encode any genes. They don't contain instructions for making proteins. Their function is purely structural. They protect the ends of chromosomes from being recognized as breaks, from being fused to other chromosomes, from degrading. Think of them as buffer zones. When a cell divides, its DNA must be copied. But the machinery that copies DNA has a quirk. It can't quite copy the very end of a linear molecule. Every time a cell divides, a little bit of the telomere is left unreplicated, effectively shortening it. For the coding regions of the genome, this would be a disaster. You'd lose genes every time a cell divided. But because telomeres are just repetitive non-coding sequences, losing a little bit each time is tolerable. The telomere gets shorter, but the actual functional genome remains intact. However, there's a limit. After enough divisions, the telomeres become critically short. When this happens, the cell interprets the shortened chromosome ends as damaged DNA, a sign that something has gone terribly wrong. In response, the cell activates a program called senescence. It stops dividing. It doesn't die immediately, but it permanently exits the cell cycle. No longer able to contribute to tissue growth or repair. This is called the Hayflick limit, named after the biologist who first observed that normal human cells can only divide a certain number of times in culture before they stop. For most human cell types, that limit is somewhere between 40 and 60 divisions. Telomere shortening is the clock that counts down those divisions. Now, this sounds like telomeres are a simple timer, a fuse that determines lifespan. And indeed, telomere length is associated with aging. Newborns have long telomeres. Old people have short ones. Across populations, shorter telomeres correlate with higher mortality risk, more age-related disease, reduced lifespan. But the relationship is not as straightforward as pop science sometimes suggests. Telomeres are not the cause of aging. They're one contributing factor among many. Here's why the story is complicated. First, not all cells are subject to the Hayflick limit. Some cells, particularly stem cells and germ cells, express an enzyme called telomerase. Telomerase adds telomeric sequences back onto chromosome ends, counteracting the shortening that happens with each division. This allows these cells to divide indefinitely without running out of telomere. Most adult cells, however, have very low or absent telomerase activity, so they do experience progressive shortening. Second, cancer cells reactivate telomerase. This is one of the hallmarks of cancer, the ability to divide indefinitely. About 85% of cancers have reactivated telomerase, allowing them to bypass the Hayflick limit and become immortal in culture. The remaining 15% use a different mechanism, called Alt, to maintain their telomeres. But the point is, unlimited cell division is dangerous. Telomere shortening is, in part, a tumor suppression mechanism. So again, we see a trade-off. Limiting cell division by allowing telomeres to shorten prevents cancer. But it also limits tissue regeneration and contributes to the depletion of functional cells. Over time. Third, telomere length is highly variable. Between individuals, between tissues in the same individual, even between cells in the same tissue. Some of this variability is genetic. Some people are born with longer telomeres than others. Some of it is environmental. Chronic stress, smoking, obesity, and inflammation are all associated with accelerated telomere shortening. Exercise, healthy diet, and good sleep are associated with longer telomeres, though the effects are modest. Fourth, while average telomere length declines with age, the shortest telomeres in a cell may be more important than the average. Even if most chromosomes have reasonably long telomeres, a few critically short ones can trigger senescence or genomic instability. This means that simple measurements of average telomere length may not capture the full picture. So, where does telomere attrition fit into the aging story? It's clearest in tissues that rely on continuous cell division. Blood cells, for example, are constantly being replenished from hematopoietic stem cells in the bone marrow. Those stem cells divide throughout life. Over decades, their telomeres shorten. Eventually, this limits their capacity to produce new blood cells, contributing to immunosenescence, the age-related decline in immune function. In the skin, cells in the basal layer divide to replace the outer layers that are constantly being shed. Telomere shortening in these cells limits the skin's ability to regenerate, contributing to thinning and reduced wound healing in old age. In the intestine, where the lining is replaced every few days, stem cells divide constantly. Telomere shortening eventually impairs this regeneration, leading to a less robust intestinal barrier and poorer nutrient absorption. But in tissues where cells rarely divide, like the brain or heart muscle, telomere shortening is less of a direct factor. Those cells face other challenges, like accumulating DNA damage that doesn't trigger division, or mitochondrial dysfunction or protein aggregation. Telomeres are less relevant there. Telomere attrition also feeds into other hallmarks. When telomeres become very short, they can become uncapped, losing the protective proteins that normally shield the chromosome end. Uncapped telomeres are recognized as DNA breaks, triggering repair pathways that can lead to chromosome fusions, deletions, and rearrangements. This exacerbates genomic instability. Cells with critically short telomeres often enter senescence. As mentioned, senescent cells secrete inflammatory factors, contributing to chronic inflammation. Another hallmark. So, telomere shortening can drive cellular senescence, which in turn drives inflammaging. Can telomeres be lengthened? Yes, experimentally. Overexpressing telomerase in mice can extend lifespan, but only if cancer is simultaneously suppressed, because telomerase also allows cancer cells to thrive. In humans, interventions that slightly slow telomere attrition, like certain lifestyle changes, show modest correlations with health. But no one has demonstrated a way to safely lengthen telomeres. Enough to produce dramatic rejuvenation. There's been a lot of hype around telomeres. Companies selling supplements or tests that claim to measure your biological age based on telomere length. Some of this is overblown. Telomere length is one biomarker among many. It's not a perfect measure of biological age, and it's not the sole determinant of lifespan. What we can say with confidence is this: telomere attrition is a real process that contributes to aging, particularly in tissues that rely on cell division. It's interconnected with genomic instability and cellular senescence. It's one thread in the larger tapestry, not the whole story, but an important part. And for you, right now, the takeaway is simple. Your telomeres are slowly shortening. This has been happening since you were born. It's a normal part of being a multicellular organism. It's one of the reasons tissues lose regenerative capacity over time. Understanding it doesn't mean you need to fear it or try desperately to stop it. It just means you see one more piece of the puzzle. Let your body rest. Let the information settle somewhere in the back of your mind. We're moving now to something even more subtle, changes not in the DNA sequence itself, but in the patterns that control how that sequence is read. Epigenetics, the third hallmark. Let's go deeper. Your genome is the same in every cell. A neuron in your brain and a cell in your liver both contain the same 3 billion base pairs of DNA, the same genes, the same instructions. And yet, a neuron and a liver cell are completely different. Different shapes, different functions, different proteins being made. How? The answer is epigenetics. The prefix epi means above or on top of. Epigenetics refers to the layers of control on top of the DNA sequence itself. The systems that determine which genes are turned on and which are turned off. Which parts of the genome are accessible and which are silenced. Think of your genome as a vast library again. But now imagine that most of the books are locked behind glass, inaccessible. Only a small subset is out on open shelves, available to be read. In a liver cell, the books about liver function are accessible. In a neuron, the books about neuron function are accessible. The rest are locked away, not because they're missing, but because they're epigenetically silenced. This control happens through several mechanisms. The most studied is DNA methylation, the addition of a small chemical group, a methyl group, to certain positions in the DNA. Heavily methylated regions are usually silenced. The genes they're not expressed. Unmethylated regions are active. The genes that are available to be transcribed into RNA and translated into proteins. Another key mechanism involves histones, the proteins around which DNA is wrapped. Chemical modifications to histones, acetylation and methylation and phosphorylation and others, change how tightly the DNA is wound. Tightly wound DNA is inaccessible, silenced. Loosely wound DNA is accessible, active. These epigenetic marks are exquisitely regulated during development. As an embryo grows, cells differentiate, committing to specific fates. A stem cell becomes a muscle cell, or a neuron, or a blood cell, and that commitment is locked in by epigenetic changes. The patterns are then maintained through cell divisions, passed from mother cell to daughter cell, preserving cell identity across a lifetime. But here's the problem. Epigenetic patterns are not perfectly stable. They drift. Over time, the precise arrangement of methylation and histone modifications becomes noisier, less crisp. Regions that should be silenced become partially active. Regions that should be active become partially silenced. The cell begins to lose its precise identity, its clarity of function. This is epigenetic alterations, the third hallmark of aging. Not changes in the DNA sequence, but changes in the instructions about how to read that sequence. Why does this happen? Several reasons, interconnected as always. First, the machinery that maintains epigenetic marks is imperfect. DNA methylation, for example, must be copied during cell division along with the DNA itself. Enzymes recognize methylated DNA and methylate the new strand to match. But this process makes errors. Over many divisions, errors accumulate, patterns drift. Second, environmental factors can alter epigenetic marks. Diet, stress, toxins. Even social experiences can lead to changes in methylation or histone modifications. Most of these changes are transient, reversed when the stimulus goes away. But some persist, especially if the exposure is chronic or happens at critical windows of development or aging. Third, DNA damage can disrupt epigenetic marks. When a DNA repair crew comes in to fix a break or a lesion, the histones in the region are displaced and the local chromatin structure is disrupted. When it's reassembled, the epigenetic marks might not be restored perfectly. Over decades of accumulated damage and repair, the epigenetic landscape becomes progressively more disordered. Fourth, certain age-related changes are programmed. Some genes are methylated with age in a highly predictable way. These changes are so consistent across individuals that they form the basis of epigenetic clocks. algorithms that can estimate biological age from the pattern of DNA methylation in a tissue sample. The first such clock was developed by Steve Horvath in 2013, and since then, many others have been developed, each refining the approach. Epigenetic clocks are remarkable. They can predict chronological age from DNA methylation with an accuracy of within a few years. More importantly, they can identify individuals whose biological age, as measured by methylation, is accelerated or decelerated relative to their chronological age. People with accelerated epigenetic age tend to have higher mortality risk, more age-related disease, poorer cognitive function. This suggests that epigenetic drift is not just a side effect of aging, but a driver. The loss of epigenetic fidelity contributes directly to cellular dysfunction. Genes involved in stress response, in metabolism, in inflammation, in DNA repair, all can be affected. A cell that has lost its epigenetic precision is a cell that's struggling to perform its role. Epigenetic alterations also interact with other hallmarks. Loss of proteostasis can be partly driven by epigenetic changes that reduce expression of chaperones and autophagy genes. Mitochondrial dysfunction can result from epigenetic silencing of genes involved in mitochondrial biogenesis. Cellular senescence can be triggered by epigenetic changes that activate senescence-associated genes. Inflammaging can be driven by hypermethylation of inflammatory genes, leading to their overexpression dot. So epigenetics is a central node in the aging network, both influenced by and influencing many other processes. Can epigenetic patterns be reset? In principle, yes. During embryonic development, there's a wave of epigenetic reprogramming that essentially erases most of the marks and resets the genome to a pluripotent state capable of becoming any cell type. This is how cloning works. You take the nucleus from an adult cell, put it in an egg, and the egg's reprogramming machinery resets the epigenome, allowing the cloned embryo to develop normally. In recent years, scientists have been exploring partial reprogramming, using factors that can reset epigenetic marks without fully reverting cells to a pluripotent state. In mice, partial reprogramming has been shown to rejuvenate tissues, restore function, and even extend lifespan in some models. But it's extremely early, and in humans, it's entirely experimental, with significant safety concerns, particularly around cancer risk. For now, what's most important is the conceptual point. Aging is not just about accumulating damage to the DNA sequence. It's also about losing the precise control over how that sequence is read. Cells become less themselves, less able to execute their specialized functions, more prone to dysfunction. This is a subtle form of damage, harder to see than a broken DNA strand, but no less consequential, and it's measurable. Epigenetic clocks are becoming important tools in aging research, allowing scientists to track biological age, to test whether interventions actually slow aging at the molecular level. As you lie here, your cells are maintaining or failing to maintain their epigenetic identities. It's happening silently, invisibly, but it's real. Part of the slow transformation from youth to age. No need to fear it. Just awareness, just understanding. Let your breathing slow. Let your thoughts drift. We're moving now from the genome itself to the products of the genome, to proteins, and the systems that keep them functional. Proteostasis, the fourth hallmark. Let's continue the journey. DNA is information. RNA is the messenger, but proteins are the workers. They're the molecules that do almost everything in a cell. Enzymes that catalyze chemical reactions. Structural components that give cells shape. Motors that transport cargo. Receptors that sense signals. Antibodies that fight infection. Hormones that coordinate between tissues. Proteins are the functional output of the genome, the molecular machines that make life work. A typical human cell contains billions of individual protein molecules, representing thousands of different types. Each protein must be folded into a precise three-dimensional shape to function. A protein's shape is determined by its amino acid sequence, the string of building blocks that's encoded in a gene and translated by the ribosome. That folding is not automatic. It's a complex, error-prone process that requires help, Inside cells are molecular chaperones, proteins whose job is to help other proteins fold correctly. They grab newly made proteins, guide them through the folding process, and release them once they've achieved their proper shape. If a protein misfolds despite this help, the chaperones try to refold it. If that fails, the protein is tagged for destruction and sent to the proteasome, a molecular shredder that breaks proteins down into amino acids, which can be recycled. This entire system, the network of chaperones, the proteasome, the quality control checkpoints, the recycling pathways, is called proteostasis, protein homeostasis, the maintenance of a healthy, functional protein population within the cell. And it works beautifully for a while. But with age, proteostasis declines. Chaperone levels drop. The proteasome becomes less efficient. Misfolded proteins accumulate. Aggregates form, clumps of tangled proteins that can't be cleared. These aggregates are toxic. They interfere with cellular processes. They can seed the formation of more aggregates, spreading like a contagion within and between cells. This is the fourth hallmark of aging, loss of proteostasis. Why does it happen? Several converging factors. First, the cellular stress response weakens with age. Young cells respond to stress, heat shock or oxidative stress or accumulation of misfolded proteins, by ramping up production of chaperones and other protective proteins. This is called the heat shock response or the unfolded protein response, depending on the context. But in old cells, this response is blunted. The sensors that detect stress still work, but the downstream amplification is reduced. So, even when there's a problem, the cell doesn't mount as strong a protective response. Second, the proteasome itself declines. Its activity, its capacity to degrade damaged or misfolded proteins, decreases with age in many tissues. The reasons are not entirely clear, but it seems to involve both a reduction in the number of active proteasomes and changes in how efficiently they work. Third, autophagy, which we'll cover in more detail in the next section, also declines. Autophagy is a broader recycling system that can engulf not just individual proteins, but entire organelles or large aggregates, break them down in acidic compartments, and return the components to the cell for reuse. When autophagy weakens, large protein aggregates that can't be handled by the proteasome persist and accumulate. Fourth, oxidative damage increases with age. Proteins are vulnerable to oxidation, which can alter their structure and function, making them more prone to misfolding and aggregation. As mitochondrial function declines, another hallmark we'll discuss later, more reactive oxygen species are produced, causing more oxidative damage to proteins. Fifth, protein synthesis itself becomes less accurate. Errors in translation, where the wrong amino acid is incorporated into a growing protein chain, increase with age. These errors produce misfolded proteins from the start, even if the gene itself is fine. The accumulation of such errors further burdens the proteostasis machinery. The consequences of lost proteostasis are widespread. In neurons, protein aggregates are a hallmark of neurodegenerative diseases. Alzheimer's disease is characterized by aggregates of beta-amyloid and tau. Parkinson's disease involves aggregates of alpha-synuclein. Huntington's disease involves aggregates of mutant huntingtin, while these diseases involve specific genetic mutations or specific proteins. The general phenomenon, the accumulation of toxic aggregates, is part of normal aging as well, just at lower levels. In muscle, loss of proteostasis contributes to sarcopenia, the age-related loss of muscle mass and strength. Damaged proteins accumulate, interfering with contractile function and triggering inflammation. In the immune system, proteostasis failure can impair the production and function of antibodies and cytokines, contributing to immunosenescence even in the absence of disease. Subtle proteostasis failure affects cellular metabolism, signaling, and resilience. A cell struggling to maintain its protein population is a cell operating below optimal capacity. And as with other hallmarks, loss of proteostasis feeds back into the system. Misfolded proteins can disrupt mitochondria, worsening mitochondrial dysfunction. They can trigger cellular senescence. As cells recognize the accumulation of damage and shut down division, they can activate inflammatory pathways, contributing to inflammaging. Is there anything that can be done? Some interventions that improve proteostasis have shown promise in animal models. Caloric restriction, which we'll discuss later, enhances proteostasis. It activates stress response pathways, increases chaperone expression, and boosts autophagy. Intermittent fasting may have similar effects. Exercise also improves proteostasis, particularly in muscle. It stimulates protein turnover, increases chaperone production, and enhances autophagy. Certain compounds can activate the heat shock response or boost proteasome activity. Some of these are being investigated as potential anti-aging therapies, though none have been proven effective in humans for slowing aging per se. And there's autophagy, which deserves its own section because it's been elevated to a separate hallmark. In the updated framework, when autophagy is functioning well, it provides a critical backup system for proteostasis, clearing out the aggregates and damaged organelles that the proteasome can't handle. But that's the next section. For now, let the idea settle. Your proteins are constantly being made, folded, used, and broken down. It's a cycle that has run flawlessly for most of your life. But it's slowing. The quality control is slipping. And the slow accumulation of damaged proteins is one more thread in the aging process. No rush. No worry. Just awareness. Breathe. Settle. We're moving now to that backup system. The cellular recycling program that clears out the big stuff. Autophagy, the fifth hallmark. Let's go on. Autophagy means self-eating. It's a cellular process that sounds almost violent, but it's actually profoundly regenerative. It's how cells break down their own components, recycle the parts, and use them to build new molecules or generate energy. There are different types of autophagy, but the most important for aging is macroautophagy, usually just called autophagy for short. In this process, a double membrane forms around a piece of the cell's interior, a damaged organelle, a clump of protein aggregates, a bit of cytoplasm. The membrane closes, forming a vesicle called an autophagosome. This autophagosome then fuses with a lysosome, an organelle filled with acidic digestive enzymes. The contents are broken down, and the building blocks are released back into the cell. Think of it as the cell's sanitation system. The proteasome is like a paper shredder, handling individual documents. Autophagy is like a garbage truck, hauling away large items that won't fit in the shredder. Autophagy runs at a low baseline level all the time, performing routine maintenance, but it can be dramatically upregulated in response to stress. Starvation is a classic trigger. When nutrients are scarce, cells ramp up autophagy to break down their own components and liberate amino acids, sugars, and fats that can be used for energy or to synthesize essential molecules. This allows the cell to survive periods of famine. Exercise triggers autophagy, particularly in muscle. The physical stress of contraction damages some cellular components, and autophagy clears out the damaged pieces, making room for new, functional ones. Even sleep involves autophagy. Brain cells, for example, increase autophagic activity during sleep, clearing out metabolic waste that accumulates during waking hours. This is one reason sleep is so critical for brain health. Autophagy is thus a key mechanism of cellular renewal. It's not just garbage disposal, it's regeneration. By breaking down and recycling damaged or unnecessary components, the cell makes space and material for new growth, maintaining a healthy, dynamic state. But autophagy declines with age. This is the fifth hallmark. The capacity to induce autophagy in response to stress decreases. The efficiency of autophagosome formation declines. The fusion of autophagosomes with lysosomes becomes less reliable. The lysosomal enzymes themselves lose activity. The whole system becomes sluggish. Why? Multiple reasons, again interconnected. Nutrient sensing pathways, which we'll discuss in the next hallmark, regulate autophagy. mTOR, a key nutrient sensor, inhibits autophagy when nutrients are abundant and activates it when nutrients are scarce. But mTOR signaling becomes dysregulated with age, often staying more active than it should be, which keeps autophagy suppressed even when it's needed. Epigenetic changes can reduce expression of autophagy genes. if the genes encoding components of the autophagic machinery are silenced or downregulated, the cell simply has less capacity to perform autophagy. Lysosomal function declines. Lysosomes are the endpoint of autophagy, and if they're not working well, the whole pathway backs up. Lysosomal dysfunction can result from accumulation of undegradable material, changes in lysosomal pH, or reduced expression of lysosomal enzymes. Oxidative damage and protein aggregates can interfere with the autophagic machinery itself. The proteins that form autophagosome, the sensors that detect cargo, the motors that move vesicles, all can be damaged or inactivated, reducing the efficiency of the system. The consequences of disabled autophagy are severe. Without efficient autophagy, damaged mitochondria accumulate, worsening mitochondrial dysfunction. Protein aggregates persist, overwhelming proteostasis. Cellular debris builds up, contributing to inflammation and senescence. In neurons. Impaired autophagy is linked to neurodegeneration. The brain relies heavily on autophagy to clear out aggregated proteins and damaged organelles. When autophagy fails, toxic aggregates accumulate, contributing to diseases like Alzheimer's and Parkinson's. In the heart, autophagy is essential for maintaining cardiac function. Impaired autophagy in aging hearts contributes to the accumulation of damaged mitochondria and proteins, leading to reduced contractility and increased risk of heart failure. In immune cells, autophagy helps control infections and regulate inflammatory responses. Disabled autophagy contributes to immunosenescence and chronic inflammation. Can autophagy be restored? Yes, to some extent. Many of the same interventions that help proteostasis also boost autophagy. Caloric restriction and intermittent fasting are among the most powerful activators of autophagy. When you eat less, or when you go longer between meals, nutrient sensors like mTOR are inhibited and autophagy is unleashed. Exercise activates autophagy, especially in muscle and brain. Certain drugs can modulate autophagy. Rapamycin, which inhibits mTOR, is a potent inducer of autophagy and has been shown to extend lifespan in mice. Metformin, a diabetes drug, may have mild autophagy activating effects through AMPK activation. Spermidine, a compound found in certain foods, has been shown to induce autophagy and extend lifespan in model organisms. But as always, what works in mice or worms doesn't automatically translate to humans. Clinical trials are ongoing, but we don't yet have proven pharmacological interventions to safely restore autophagy in aging. Humans. What we do have is lifestyle. Fasting, or at least not eating constantly. Physical activity, sleep, these are accessible, safe ways to support autophagy. And understanding the process itself brings a kind of peace. Your body has this incredible system for self-renewal, a way to break down the old and build the new. It's been working your whole life. It's slowing now, but it's not gone. And you can support it gently with how you live. Let the idea rest. Let your body rest. Autophagy might even be happening right now in some small way. As you lie still in the dark, we're moving to the next hallmark, a broader one, about how your cells sense the world and decide what to do with resources, nutrient sensing. The sixth hallmark. Let's continue. Every cell faces a fundamental decision, moment to moment, grow or maintain, build new structures or repair existing ones, divide or stay quiescent. These decisions are made by nutrient-sensing pathways, molecular circuits that detect the availability of food, energy, and building blocks, and adjust cellular behavior accordingly. When nutrients are abundant, the signal is grow, synthesize proteins, replicate DNA, prepare to divide, invest in the future. When nutrients are scarce, the signal is conserve, activate repair, boost autophagy, slow down unnecessary processes, survive until conditions improve. This makes evolutionary sense. In a feast, it's advantageous to grow and reproduce. In a famine, it's advantageous to hunker down and maintain what you have until the feast returns. The problem is that in modern life, for most people in developed countries, nutrients are never scarce. We exist in a permanent state of feast. and our nutrient-sensing pathways, optimized for a world of variable food availability, are constantly signaling grow, grow, grow. This chronic growth signaling has costs. Growth and maintenance are, to some extent, in tension. Resources devoted to making new proteins are not available for repairing damaged ones. Pathways that promote cell division can conflict with pathways that promote longevity, and over time, the nutrient-sensing pathways themselves become dysregulated. They stop responding appropriately to signals. They get stuck in growth mode even when maintenance would be more appropriate. This is the sixth hallmark of aging, deregulated nutrient sensing. Let's look at the main players. MTOR stands for mechanistic target of rapamycin, named after a drug that inhibits it. mTOR is a master regulator of growth. When amino acids and other nutrients are abundant, mTOR is active, promoting protein synthesis, ribosome production, and cell growth. It also inhibits autophagy because if resources are plentiful, there's no need to recycle. But when mTOR is too active for too long, problems arise. Chronic mTOR activation drives cellular senescence in some contexts. It impairs autophagy, allowing damaged components to accumulate. It increases protein synthesis even when quality control is already overwhelmed, worsening proteostasis. It promotes inflammation. In aging, mTOR signaling tends to remain elevated, particularly in response to nutrients. The pathway becomes less sensitive to inhibition, less capable of shutting down when it should. Rapamycin, which inhibits mTOR, is one of the most robust lifespan-extending interventions in animal models. Mice treated with rapamycin live longer, have less cancer, better immune function. IMPR-OVED metabolism. The effect is seen even when treatment starts late in life, but rapamycin is an immunosuppressant, used clinically to prevent organ transplant rejection. Long-term use has side effects. So, while it's a powerful tool for research, it's not yet a proven or approved anti-aging therapy for healthy humans. Insulin and IGF-1 signaling is another major nutrient-sensing pathway. Insulin is released when blood sugar rises, signaling cells to take up glucose. IGF-1, insulin-like growth factor 1, is a growth hormone that promotes cell division and growth throughout the body. Together, the insulin-IGF-1 signaling pathway, often abbreviated IIS, promotes growth and metabolism, but chronic elevation of this pathway, as happens in obesity, insulin resistance, and type 2 diabetes, is associated with accelerated aging, increased cancer risk, and reduced lifespan. Conversely, reduced IIS signaling is associated with longevity. Genetic mutations that reduce IGF-1 levels or insulin signaling have been found in several long-lived populations. In model organisms, worms and flies with reduced IIS live much longer than normal. In humans, the picture is more complex. Very low IGF-1 is not clearly beneficial and can have downsides, but chronically high levels, especially in the context of metabolic disease, are clearly harmful. AMPK, AMP-activated protein kinase, is a sensor of cellular energy status. When energy is low, when ATP, the cell's energy currency, is depleted, AMPK is activated. It responds by shutting down energy-consuming processes like protein synthesis and turning on energy-generating processes like autophagy and mitochondrial. BIOGENESIS.AMPK is essentially a low battery alarm. It shifts the cell into conservation mode. With age, AMPK activity tends to decline. Cells become less able to respond to energy stress, less able to activate protective programs when needed. Metformin, the diabetes drug, activates AMPK. This is thought to be one reason metformin has been associated with health benefits beyond glucose control, including possible reductions in cancer risk and overall mortality. There's an ongoing clinical trial, the TAME trial, targeting aging with metformin, designed to test whether metformin can delay aging-related diseases in non-diabetic older adults. Sirtuins are another family of nutrient sensors, particularly sensitive to NAD+, a molecule involved in cellular energy metabolism. Sirtuins regulate gene expression, DNA repair, and mitochondrial function. They're activated by caloric restriction and are thought to mediate some of its beneficial effects. NAD+ levels decline with age, which may reduce sirtuin activity. Boosting NAD+ through supplementation with precursors like NMN or NR has been a popular area of research and commercial interest. Though the human evidence for anti-aging benefits remains limited, all of these pathways, mTOR, IAS, AMPK, sirtuins are interconnected. They crosstalk, regulate each other, and collectively determine whether a cell prioritizes growth or maintenance, division or repair, accumulation or recycling. And the central insight of aging research is this: chronically activating growth pathways accelerates aging. Periodically activating maintenance pathways by caloric restriction, fasting, exercise, or certain drugs may slow aging. This doesn't mean starvation is good. Severe malnutrition is catastrophic, but mild intermittent restriction, enough to shift the balance from constant growth toward more maintenance, appears to be beneficial across a huge range of organisms. In humans, caloric restriction has been tested in controlled studies. The calorie trials showed that 2 years of moderate caloric restriction improved markers of aging, reduced inflammation, improved metabolic health. But the trials weren't long enough or large enough to prove lifespan extension, and adherence to long-term caloric restriction is difficult. Intermittent fasting, time-restricted eating, and other dietary patterns that reduce overall calorie intake or extend the time between meals may have similar benefits, though again, the human evidence is preliminary. The broader point is that how you eat, and how often you eat, influences these nutrient-sensing pathways. Constant snacking, high sugar and refined carbs, overeating, all drive IAS and mTOR in ways that may accelerate aging. Longer fasting windows, lower glycemic foods, moderate protein intake, all may shift the balance toward maintenance. But this isn't medical advice. It's biology. The specific choices that are right for any individual depend on countless factors: health status, activity level, goals, preferences. The science just tells us that these pathways exist, that they matter, and that they can be influenced, and deregulation of these pathways, the loss of appropriate responsiveness, the chronic skew toward growth signaling is one of the core features of aging. Not the only one, but an important one, connected to many others. Rest with that thought. Your cells are sensing, deciding, adjusting constantly. It's a dance that's gone on every second of your life. And understanding it, even a little, brings you closer to the mechanisms that shape health and longevity. Breathe slowly. Let your mind drift. We're moving now to the powerhouses of the cell, the organelles that produce energy and so much more. Mitochondria, the seventh hallmark. Let's continue. Inside almost every cell in your body are hundreds or thousands of mitochondria, tiny organelles that are, in a very real sense, the descendants of ancient bacteria. About 2 billion years ago, a bacterium capable of aerobic respiration was engulfed by a larger cell. Instead of being digested, it survived, forming a symbiotic relationship. The host cell provided protection and nutrients. The bacterium provided energy. Over eons, the bacterium evolved into the mitochondrion, losing most of its independent genes but retaining a small genome and the ability to produce ATP, the universal energy currency of life. Mitochondria are thus both part of us and separate from us. They have their own DNA, a small circular genome inherited exclusively from the mother. They replicate independently of the cell cycle, and they're essential. Without them, complex multicellular life as we know it wouldn't exist. Their primary job is to produce ATP through a process called oxidative phosphorylation. They take sugars and fats, break them down, and use the energy to pump protons across a membrane, creating a gradient. That gradient drives a molecular turbine, ATP synthase, which cranks out ATP. But mitochondria do much more than produce energy. They're involved in calcium signaling, regulation of apoptosis, cell death, synthesis of key metabolites, and they generate reactive oxygen species, ROS, which despite their reputation as damaging, also serve as important signaling molecules, Mitochondrial health is thus central to cellular health, and mitochondrial dysfunction is central to aging. The seventh hallmark is mitochondrial dysfunction, the decline in mitochondrial efficiency, integrity, and number with age. What goes wrong? Several things, interconnected as always. First, mitochondrial DNA, mtDNA, accumulates mutations. It's particularly vulnerable. mtDNA sits right next to the inner membrane where oxidative phosphorylation happens, exposed to high levels of ROS, and mtDNA has fewer protective histones than nuclear DNA, less robust repair mechanisms. Over time, mutations accumulate. Most are harmless or mildly deleterious, but some impair the function of the proteins involved in ATP production or ROS management. A cell typically has hundreds of mitochondria, each with multiple copies of mtDNA. So for a mutation to matter at the cellular level, it often has to reach a threshold, where a significant fraction of the mtDNA copies carry the mutation. This can happen through a process called clonal expansion, where a mitochondrion with a mutant genome replicates more than others, gradually taking over the population within the cell. When enough mtDNA is mutated, the cell's energy production drops. It becomes less able to meet metabolic demands. It produces more ROS, creating a vicious cycle of damage. Second, mitochondrial dynamics. The processes of fusion and fission become impaired. Mitochondria are not static. They constantly fuse together and divide, mixing their contents, allowing damaged components to be diluted and functional ones to be shared. This dynamic behavior is essential for quality control, but with age, the balance shifts. Fission increases, fusion decreases. Mitochondria become fragmented. Fragmented mitochondria are less efficient and more prone to producing ROS. Third, mitophagy. The selective autophagy of damaged mitochondria declines. When a mitochondrion is damaged to function, it's supposed to be marked for destruction and engulfed by an autophagoson. This prevents the damaged mitochondrion from poisoning the cell. But as autophagy declines with age, damaged mitochondria persist, accumulating in tissues. Fourth, mitochondrial biogenesis, the creation of new mitochondria, slows. This is regulated by nutrient-sensing pathways and transcription factors like PGC1-alpha. With age, the signals that promote mitochondrial biogenesis weaken, and cells produce fewer new mitochondria to replace old ones. The result is that cells end up with fewer, more damaged mitochondria, producing less ATP and more ROS. This creates cellular energy deficits, particularly in tissues with high energy demands like the brain, heart, and muscle. In the brain, mitochondrial dysfunction contributes to neurodegeneration. Neurons are extremely energy demanding. When mitochondria fail, neurons can't maintain their ion gradients, can't support synaptic transmission, become vulnerable to excitotoxicity and cell death. In the heart, mitochondrial dysfunction reduces contractile force. The heart muscle, which beats continuously, relies on a constant supply of ATP. Impaired mitochondria mean less ATP, weaker contractions, higher risk of heart failure. In muscle, mitochondrial dysfunction contributes to sarcopenia and exercise intolerance. Muscles lose mass and strength, fatigue more easily, and mitochondrial ROS, when produced in excess, damage proteins, lipids, and DNA, contributing to proteostasis failure, genomic instability, and inflammation. So mitochondrial dysfunction feeds into multiple other hallmarks. There's also the theory that aging is fundamentally a mitochondrial disease, that the accumulation of mtDNA damage and mitochondrial dysfunction is the primary driver of all other aging processes. This is called the mitochondrial free radical theory of aging, first proposed in the 1970s. It's been influential, but it's also been revised. It's now clear that ROS are not purely damaging. They're also signals, and antioxidant supplementation, which might be expected to slow aging if ROS were the sole problem, has largely failed in clinical trials. So mitochondrial dysfunction is important, but it's not the whole story. It's one hallmark among many, interconnected with the rest. Can mitochondrial function be improved? Some evidence suggests yes. Exercise is one of the most powerful interventions. It stimulates mitochondrial biogenesis, increases mitochondrial density, improves efficiency. Even in older adults, exercise training can improve mitochondrial function, increase ATP production, reduce ROS production. Caloric restriction enhances mitochondrial efficiency and biogenesis, partly through AMPK and sirtuin activation, NAD boosting, through supplements like NMN or NR, has been shown in animal studies to improve mitochondrial function, though human data is still limited. Certain mitochondrial-targeted antioxidants, like Mitoc, which deliver antioxidants specifically to mitochondria, have shown some promise in animal studies and early human trials, though results are mixed, and there's emerging interest in mitochondrial transplantation or gene therapy. Delivering healthy mitochondria or correcting mtDNA mutations. These are far from clinical reality but represent future possibilities. For now, the message is clear. Mitochondria matter. They're not just batteries. They're signaling hubs, metabolic centers, quality control nodes. And their decline is a core feature of aging. Take a deep breath. Feel your body using oxygen right now, burning fuel, producing energy in trillions of mitochondria. They've been doing this your whole life, and they'll keep doing it as long as you live. We're moving now to a particularly fascinating hallmark, one that involves cells that stop dividing but don't die, instead sitting in tissues and causing trouble. Cellular senescence. The eighth hallmark. Let's go deeper. Cells have choices. When faced with stress or damage, they can try to repair. They can divide, diluting the damage between daughter cells, or they can die via apoptosis, removing themselves quietly without causing inflammation. But there's a fourth option, and it's both protective and problematic. They can become senescent. Senescence is a state where a cell permanently stops dividing but doesn't die. It's a stable, long-lasting state, distinct from quiescence, where cells can re-enter the cell cycle if conditions change. Senescent cells are locked out. They will never divide again. This was first observed in the 1960s by Leonard Hayflick, who noticed that normal human cells stopped dividing after a certain number of divisions in culture. At the time, it was seen as a curiosity, maybe an artifact of cell culture. But over decades, it became clear that senescence is a real, important phenomenon in living organisms. Why would a cell become senescent? It's actually a protective mechanism most of the time. If a cell accumulates serious DNA damage that can't be fully repaired, the safest response is often to stop dividing, because division would pass the damage on to daughter cells, potentially creating cells with dangerous mutations, cells that might become cancerous. Senescence is thus a form of tumor suppression. Similarly, if a cell's telomeres become critically short, signaling that it's reached the end of its replicative lifespan, senescence prevents it from continuing to divide and risking genomic catastrophe. Senescence can also be triggered by oncogenes, genes that promote cell growth and division. If an oncogene becomes hyperactive, the cell recognizes this as dangerous and shuts down, entering senescence rather than progressing toward cancer. So senescence is a safeguard. It's the body's way of neutralizing potentially dangerous cells, without the immediate need to destroy them. And in youth, senescence is even beneficial in certain contexts. During wound healing, cells at the injury site become senescent, secreting factors that recruit immune cells, promote tissue remodeling, and then, crucially, the senescent cells are cleared away by the immune system once the healing is done. In embryonic development, senescence plays a role in tissue patterning and organ formation. The problem is that with age, senescent cells accumulate. The rate of cells entering senescence increases, driven by the accumulation of DNA damage, telomere attrition, mitochondrial dysfunction, and other stressors. And simultaneously, the efficiency of the immune system in clearing senescent cells declines. So, senescent cells build up in tissues. And this is where the trouble starts, because senescent cells, while they don't divide, are far from quiet. They secrete a cocktail of inflammatory cytokines, growth factors, and proteases, collectively called the senescence-associated secretory phenotype, or SOSP, the SASP is, in the short term, part of the beneficial function. It recruits immune cells to clear the senescent cells. It promotes wound healing and tissue remodeling. But when senescent cells persist, the chronic SASP becomes harmful. The inflammatory cytokines spread inflammation to neighboring cells, creating a local inflammatory environment. This inflammation can damage nearby healthy cells, trigger more senescence, impair tissue function, and contribute to age-related diseases. The proteases secreted by senescent cells degrade the extracellular matrix, the structural scaffold of tissues, leading to tissue weakening and loss of architecture. The growth factors can stimulate abnormal cell growth, potentially promoting cancer in nearby cells that haven't fully transformed yet. Senescent cells also alter the stem cell niche, the environment where stem cells reside. The SASP can impair stem cell function, contributing to stem cell exhaustion and reduced tissue regeneration. So senescence, protective in the moment, becomes a driver of aging when cells accumulate chronically. This is the 8th hallmark. Senescent cells have been found in almost every aged tissue: skin, fat, liver, kidney, lung, blood vessels, brain. They're relatively rare, typically less than a few % of the total cell population, but their impact is outsized because of the SASP. In mice, genetic approaches that selectively kill senescent cells lead to dramatic improvements. Mice live longer, have less cancer, better kidney function, healthier hearts, less frailty. Removing senescent cells essentially rejuvenates tissues. This has led to huge interest in Senolytics, drugs that selectively kill senescent cells. The idea is to develop compounds that can identify and eliminate these troublesome cells, allowing tissues to function more youthfully. Several senolytic compounds have been identified. Dasatinib and quercetin, often used in combination, are among the most studied. Dasatinib is a cancer drug, a tyrosine kinase inhibitor. Quercetin is a plant flavonoid found in foods like onions and apples. Together, they seem to induce apoptosis in senescent cells, though the mechanism isn't entirely clear. In early human trials, dasatinib plus quercetin has shown some effects. In patients with idiopathic pulmonary fibrosis, a lung disease associated with senescence, the combination improved physical function and reduced senescent cell markers. In small studies of older adults, it reduced inflammatory markers and improved vascular function. But these are very early results. The trials are small, short-term, and mostly in diseased populations, not healthy aging. Long-term safety is unknown, and it's not clear whether intermittent senolytic treatment would be effective or necessary, or how often it would need to be given. Other senolytics are in development, more specific, potentially safer. But as of now, there's no approved senolytic therapy for aging. There are also approaches to suppress the SASP without killing senescent cells, called xenomorphics. These might reduce the inflammatory secretions, making senescent cells less harmful even if they remain present. For now, lifestyle factors may matter. Exercise has been shown to reduce senescent cell burden in some tissues, probably through improved immune clearance. Healthy diet, avoiding obesity and chronic inflammation, may also reduce the rate of senescence accumulation. And it's worth emphasizing, senescence is not purely bad. It's a nuanced, context-dependent phenomenon. In the right place at the right time, it's protective. It's only when cells persist chronically, in the wrong place at the wrong time, that it becomes harmful. This is a theme in aging. Many protective mechanisms have a dark side when chronic or dysregulated. Senescence, inflammation, stress responses all follow this pattern. Let the complexity settle. Senescent cells, once heroes, become villains when they refuse to leave the stage. It's almost a tragedy in the classical sense. Good intentions, unintended consequences. Breathe. Rest. We're moving to the 9th hallmark, about the cells that should be heroes, that should be regenerating tissues, but are running out of steam. Stem cell exhaustion. Let's continue. Your body is constantly renewing itself. The lining of your gut is replaced every few days. Your skin sheds and regenerates continuously. Blood cells are replenished from the bone marrow at a rate of hundreds of billions per day. Muscle and other tissues repair damage and replace worn-out cells. This renewal is possible because of stem cells. Stem cells are special cells that can divide to produce more stem cells, self-renewal, and also differentiate into specialized cell types. The functional cells of tissues. There are different kinds of stem cells. Embryonic stem cells are pluripotent, capable of becoming any cell type in the body. But in adults, stem cells are more restricted, found in specific tissues, capable of producing only the cell types of that tissue. These are called adult stem cells or tissue-specific stem cells. The blood system is maintained by hematopoietic stem cells in the bone marrow. The intestine has stem cells at the base of the crypts in the intestinal lining. Skeletal muscle has satellite cells that activate to repair muscle fibers. The brain has neural stem cells, though their role and abundance are more limited than in other tissues. Even the skin has stem cells in hair follicles and the basal layer. These stem cells are the foundation of tissue regeneration. They allow the body to heal, to replace what's lost, to maintain function despite constant wear and tear. But with age, stem cell function declines. This is the 9th hallmark, stem cell exhaustion. What does this mean? Several changes happen. First, stem cell number can decline. In some tissues, stem cells are simply depleted over time. They divide, producing differentiated progeny, but the self-renewal component isn't perfect. A little bit of the stem cell pool is lost with each round of division. Over decades, the pool shrinks. Second, stem cell quality declines. Even if the number is maintained, individual stem cells become less functional. They divide more slowly. They differentiate less efficiently. They produce progeny that are themselves compromised. Third, the stem cell niche, the local environment that supports stem cells, deteriorates. Stem cells rely on signals from surrounding cells, from the extracellular matrix, from circulating factors. As those elements change with age, the niche becomes less supportive, and stem cells become quiescent or dysfunctional even if they're intrinsically capable of function. Why does this happen? Stem cells are subject to all the same aging processes as other cells. They accumulate DNA damage. Their telomeres shorten, especially in tissues where stem cells divide frequently. Their epigenetic patterns drift. Their mitochondria become dysfunctional. Senescent cells in the niche secrete SASP factors that inhibit stem cell activity. So stem cell exhaustion is not a separate, independent process. It's an integrative consequence of the other hallmarks: genomic instability, telomere attrition, epigenetic alterations, mitochondrial dysfunction, cellular senescence. All converge to impair stem cell function. The consequences are visible in aging tissues. In the blood system, hematopoietic stem cell exhaustion leads to reduced production of immune cells, contributing to immunosenescence, the ability to respond to infection, to generate diverse antibodies to clear pathogens. All decline. There's also an increased risk of blood cancers, as accumulated mutations in aging stem cells can lead to clonal hematopoiesis, the expansion of mutant clones that may eventually progress to leukemia. In the intestine, declining stem cell function reduces the regenerative capacity of the gut lining. This can lead to impaired nutrient absorption, increased intestinal permeability, and greater susceptibility to gastrointestinal diseases. In muscle, satellite cell exhaustion contributes to sarcopenia. Muscle injuries heal more slowly. Muscle mass declines. Strength and function are lost. In the skin, stem cell decline leads to slower wound healing, thinner skin, less hair regeneration, visible signs of aging. Even in the brain, where the role of stem cells is more limited, the decline in neurogenesis, the production of new neurons in certain brain regions, is associated with cognitive decline and mood disorders. Can stem cell function be restored? To some extent, yes. In mice, rejuvenating the stem cell niche has been shown to restore function. For example, exposing old stem cells to a youthful environment, through parabiosis experiments where the blood of young and old mice is shared, can partially restore stem cell activity. This suggests that at least some of the decline is niche-driven, not purely intrinsic to the stem cells themselves. Clearing senescent cells from tissues can improve the niche and enhance stem cell function. Certain interventions, Like exercise, stimulates stem cell activity. Exercise promotes muscle satellite cell activation, enhances neurogenesis, improves hematopoietic stem cell function. Caloric restriction has been shown to preserve stem cell number and function in some contexts, possibly by reducing metabolic stress and enhancing autophagy. There's also interest in stem cell therapies, transplanting young stem cells into aged tissues to replenish the pool. This is being explored clinically for various conditions, but it's complex and not yet a standard treatment for aging. And partial reprogramming, which we'll touch on later, has shown some ability to rejuvenate aged stem cells, restoring markers of youthfulness and improving function. But again, these are all experimental. For now, the main message is that stem cell exhaustion is a critical component of aging, limiting the body's ability to repair and regenerate. And it's deeply interconnected with the other hallmarks, both a cause and a consequence. The body you have today is not the body you had 10 years ago. Most of the cells have been replaced, renewed, regenerated. But the system that does that renewal is wearing down. And that's part of aging. No fear. Just awareness. Just the quiet recognition of how it works. Rest. Sink deeper. We're moving now to a hallmark about communication, about how cells talk to each other and how that conversation becomes garbled with age. Intercellular communication, the 10th hallmark. Let's continue. No cell is an island. Every cell in your body exists in constant communication with other cells, near and far. Cells secrete signaling molecules, cytokines, hormones, growth factors, that travel to neighbors or through the bloodstream to distant tissues. They receive signals through receptors on their surface, triggering cascades of responses inside. They coordinate, cooperate, respond to the needs of the whole organism. This communication is essential for homeostasis, the maintenance of stable internal conditions despite external changes. It's how your immune system coordinates to fight infections, how your metabolism adjusts to food intake, how tissues repair after injury, how organs work together to support life. But with age, communication becomes noisy. Signals are garbled. Responses are exaggerated or blunted. The coordination breaks down. This is the 10th hallmark, altered intercellular communication. One of the most important aspects of this is inflammaging, a term coined to describe chronic, low-grade inflammation that increases with age even in the absence of infection or acute disease. Inflammation in the acute setting is good. It's how the body responds to infection or injury. Immune cells are recruited. Pathogens are attacked. Damaged tissue is cleared. Then, once the job is done, inflammation resolves and homeostasis is restored. But in aging, inflammation doesn't resolve. It persists, not at the intense level of an infection, but at a low, smoldering level, continuously present, gradually causing damage. Where does this chronic inflammation come from? Multiple sources, or interconnected. Senescent cells secrete SASP factors, which include inflammatory cytokines like IL-6, IL-1 beta, TNF-alpha. These spread inflammation locally and, if senescent cells are widespread, systemically. Damaged mitochondria release damage-associated molecular patterns, DAMPs, that trigger inflammatory responses. mtDNA, for instance, can leak out of damaged mitochondria and activate innate immune receptors, causing inflammation, cellular debris from impaired autophagy and proteostasis, accumulates and activates inflammasomes, multi-protein complexes that trigger the release of inflammatory cytokines. The gut microbiome changes with age, often shifting toward less beneficial compositions. Dysbiosis, which we'll discuss more in the next section. A disrupted microbiome can lead to increased intestinal permeability, leaky gut, allowing bacterial products like lipopolysaccharide to enter the bloodstream and trigger systemic inflammation. The immune system itself ages, a process called immunosenescence. T cells and B cells become less diverse, less able to respond to new pathogens. But at the same time, innate immune cells become hyperactive, responding more aggressively to perceived threats even when there's no actual infection. This creates a paradox: weaker adaptive immunity, stronger but misdirected innate inflammation. Adipose tissue, fat, becomes more inflammatory with age, especially visceral fat. Fat cells secrete cytokines and adipokines. In aging, the profile shifts toward more inflammatory signals. All of these sources contribute to elevated circulating levels of inflammatory markers. CRP, IL-6, TNF-alpha. These are measurable in blood and consistently elevated in older adults compared to younger ones. And this chronic inflammation is not benign. It contributes to almost every age-related disease. Cardiovascular disease, where inflammation promotes atherosclerosis, plaque formation in arteries. Alzheimer's disease, where neuroinflammation exacerbates neurodegeneration. Diabetes, where inflammation impairs insulin signaling. Cancer, where chronic inflammation can promote tumor growth and metastasis. Osteoporosis, arthritis, frailty, all have inflammatory components. Inflammaging is thus a central, integrative feature of aging. It's both a consequence of other hallmarks and a driver of further decline. Beyond inflammation, intercellular communication changes in other ways. Hormonal signaling declines. Growth hormone and sex hormones, estrogen, testosterone, drop with age, affecting metabolism, muscle mass, bone density, mood. Cognitive function, neuroendocrine signaling. The communication between the brain and the rest of the body becomes less precise. The hypothalamic pituitary adrenal axis, which regulates stress responses, becomes dysregulated, often overactive, contributing to chronic cortisol elevation. Immune surveillance declines. The ability to detect and clear cancer cells, infected cells, senescent cells, all weaken. This allows these problematic cells to persist and accumulate. Even the signaling within the extracellular matrix, the structural network surrounding cells, changes. Matrix becomes stiffer, less elastic, altering mechanical signals that cells receive, affecting everything from stem cell behavior to tissue repair. Can altered intercellular communication be corrected? Partially, perhaps. Reducing senescent cells reduces SASP, lowering inflammation. Improving mitochondrial function reduces damp release, supporting gut health through diet. Probiotics, prebiotics may reduce dysbiosis and leaky gut, lowering systemic inflammation. Exercise has broad anti-inflammatory effects, reducing circulating inflammatory markers and improving immune function. Caloric restriction and fasting reduce inflammation, partly by reducing oxidative stress and improving autophagy. Certain drugs have anti-inflammatory effects. Low-dose aspirin, statins, metformin, all have been studied for their potential to reduce inflammaging, with mixed results. And there's interest in targeting specific inflammatory pathways with biologics, drugs that block specific cytokines like IL-6 or TNF-alpha. These are used clinically for autoimmune diseases. Whether they could be used safely and effectively to reduce inflammaging in general aging populations is an open question. For now, the key insight is that aging is not just about what happens inside cells. It's about how cells communicate or fail to communicate. It's about the breakdown of coordination, the loss of precision in the signals that maintain health. And inflammation, chronic, low-grade, persistent, is the clearest example. A signal meant to protect, meant to heal, stuck in the wrong position, slowly causing harm. Let that settle. Your body is full of signals right now. Cytokines, hormones, neurotransmitters, flowing, binding, triggering, constantly. And the pattern of those signals is shifting slowly. With time, breathe, rest. We're moving now to a relatively new addition to the hallmarks, one that connects aging to the trillions of microbes living inside you. Dysbiosis, the 11th hallmark. Let's go on. You are not alone. Even in isolation, even in the deepest solitude, you carry with you a vast, invisible population. Trillions of bacteria, archaea, fungi, viruses, collectively called the microbiome, living on your skin, in your mouth, in your gut. The gut microbiome is the densest and most studied. Your large intestine is home to more microbial cells than you have human cells in your entire body. These microbes are not passive passengers. They're metabolically active, producing vitamins. breaking down complex fibers that your own enzymes can't digest, training your immune system, even producing neurotransmitters that affect mood and brain function. The relationship is symbiotic. You provide them with a warm, nutrient-rich environment. They provide you with metabolic capabilities and immune support. It's an ancient partnership refined over millions of years of coevolution. But the microbiome is not static. It changes across the lifespan. Infants have a relatively simple microbiome, dominated by species acquired from the mother and environment. By adulthood, the microbiome is more diverse, more stable. A complex ecosystem dominated by two major bacterial phyla, Bacteroidetes and Firmicutes. And then, with age, it changes again. This is dysbiosis, the 11th hallmark of aging, recently added to the framework to reflect growing evidence that the microbiome plays a significant role in aging. What characterizes age-related dysbiosis? Several trends have been observed. Diversity decreases. A diverse microbiome is generally considered healthy, with many different species coexisting, providing functional redundancy and resilience. In older adults, especially those who are frail or institutionalized, microbial diversity declines. Certain species dominate, others disappear. The balance shifts. The ratio of beneficial to potentially harmful bacteria changes. Commensal bacteria, ones that are neutral or beneficial, decline. Opportunistic pathogens, bacteria that can cause inflammation or infection if given the opportunity, increase. Beneficial metabolites decline. Certain gut bacteria produce short-chain fatty acids, SCFA letters, like butyrate, acetate, propionate, by fermenting dietary fiber. SCFA letters are absorbed by the gut lining and used as fuel. They also have anti-inflammatory effects, support gut barrier function, and influence metabolism. In aging, SCFA production often declines, partly because the bacteria that produce them are less abundant. Pro-inflammatory species increase. Some bacteria produce metabolites or components, like lipopolysaccharide, that trigger inflammatory responses. In aged microbiomes, these species can become more prevalent. Why does the microbiome change with age? Several factors contribute. Diet changes. Older adults may eat less fiber, less diverse foods, more processed foods, all of which affect the microbiome medications. Older adults are more likely to be on multiple medications. Antibiotics directly kill bacteria, disrupting the microbiome. But other drugs, like proton pump inhibitors for acid reflux or metformin, also alter microbial composition, reduced mobility and social interaction. People who are less active, who are institutionalized, tend to have less diverse diets and environments, which correlates with less diverse microbiomes. Changes in the gut itself. The gut lining changes with age. Transit time can slow. Digestive enzyme production declines. The immune cells in the gut mucosa change, becoming less tolerant, more inflammatory. All of this affects the microbial environment, and there are likely bidirectional effects. Dysbiosis affects the host, and host aging affects the microbiome, creating feedback loops. The consequences of dysbiosis are increasingly recognized. A disrupted microbiome can contribute to chronic inflammation, partly by increasing intestinal permeability. A healthy gut lining is a tight barrier, allowing nutrients through but keeping bacteria and bacterial products out of the bloodstream. But when the lining is compromised, leaky gut, bacterial fragments enter circulation and trigger immune responses, contributing to inflammaging. Dysbiosis can impair nutrient absorption and metabolism, contributing to frailty and malnutrition in older adults. It can affect immune function. The gut microbiome trains the immune system. Dysbiosis is associated with weaker vaccine responses, increased infection risk, and chronic inflammation. There's even evidence linking the gut microbiome to brain health. The gut-brain axis. Microbial metabolites can influence neuroinflammation, neurotransmitter production, and mood. Dysbiosis has been associated with depression, cognitive decline, and neurodegenerative diseases. Though the mechanisms are still being worked out, can the microbiome be rejuvenated? Partially, perhaps, diet is the most direct intervention. Increasing fiber intake, eating diverse plant foods, fermented foods like yogurt, kimchi, sauerkraut, all can shift the microbiome toward a healthier composition. Probiotics, live beneficial bacteria, can help, though effects are often strain-specific and individual-dependent. Not every probiotic works for everyone. Prebiotics, non-digestible fibers that feed beneficial bacteria, can promote the growth of SCFA-producing species, fecal microbiota transplantation, FMT, transferring stool from a healthy donor to a recipient, can profoundly alter the microbiome. It's used clinically for severe Clostridioides difficile infections with great success. Whether it could be used to rejuvenate the aged microbiome is an area of active research, with some promising early results in animal models. Exercise also influences the microbiome, promoting diversity and SCFA production. The field is young, and much is still uncertain, but the inclusion of dysbiosis as a hallmark reflects the recognition that aging is not just about the human cells in your body. It's about the entire ecosystem, the holobiont, the collective of human and microbial cells that function as one. You are a walking, breathing ecosystem, and the health of that ecosystem matters for the health of the whole. Let that image settle. Trillions of tiny lives coexisting with yours, affected by how you live, affecting how you age. It's humbling, and it's beautiful in a way. Rest. Breathe. We're moving to the 12th and final hallmark, one that ties together much of what we've discussed. The chronic activation of the immune system in ways that do more harm than good. Chronic inflammation and immune aging. Let's finish the map. The immune system is your body's defense force. It patrols for invaders, bacteria, viruses, fungi, parasites. It clears out damaged cells and debris. It monitors for cancer, cells that have become abnormal and dangerous. It's extraordinarily complex, with dozens of cell types, multiple layers of defense, exquisite specificity and memory. When you're young, your immune system is robust. It responds rapidly to new infections. It generates diverse antibodies. It clears pathogens efficiently. It resolves inflammation when the threat is gone. It maintains tolerance, not attacking your own tissues. But with age, the immune system changes, not in one direction, but into seemingly contradictory directions simultaneously. It becomes weaker in some ways and overactive in others. This is immunosenescence and inflammaging, aspects of the 12th hallmark, chronic inflammation and immune aging. Let's start with the weakening, the adaptive immune system, which includes T cells and B cells, declines. This part of the immune system is responsible for specific, targeted responses to pathogens and for immunological memory. The reason vaccines work and why you don't get the same infection twice. T cells are produced in the thymus, an organ in your chest, but the thymus atrophies with age, shrinking and producing fewer new T cells. By middle age, thymic output is a fraction of what it was in childhood. The T cells you have also change. The naive T cell pool, T cells that haven't yet encountered their specific antigen, shrinks. The memory T cell pool, T cells that have responded to past infections, expands. This shift means the immune system becomes increasingly focused on old threats and less able to respond to new ones. B cells, which produce antibodies, also decline in function. They produce fewer antibodies in response to new antigens. The antibodies they do produce are often less specific, less effective. Vaccine responses weaken with age, one of the most clinically evident signs of immunosenescence. The innate immune system, the first line of defense involving macrophages. Neutrophils, natural killer cells, also changes. But here, the change is not simple decline. These cells often become hyperactive, but in a misdirected way. They're slower to respond to actual infections, but they're more prone to producing inflammatory cytokines in response to non-threatening stimuli. This creates the paradox: weak defense, strong inflammation. Older adults are more susceptible to infections. Flu, pneumonia, urinary tract infections, all are more common and more severe. At the same time, baseline inflammation is elevated, contributing to chronic diseases. This chronic inflammation is not caused by infection. It's sterile inflammation driven by endogenous signals. Senescent cells, damaged mitochondria, cellular debris, dysbiosis all contribute, as we've discussed, and the aging immune system itself contributes, losing regulatory control, losing the ability to turn off inflammatory signals when they're no longer needed. Regulatory T cells, a subset of T cells that suppress immune responses and maintain tolerance, decline in function. This can lead to increased autoimmunity, where the immune system mistakenly attacks the body's own tissues. Autoimmune conditions, rheumatoid arthritis, thyroiditis, others become more common with age. Inflammaging also affects the blood-brain barrier and the brain's resident immune cells, microglia. Chronically activated microglia contribute to neuroinflammation, which is implicated in Alzheimer's, Parkinson's, and general cognitive decline. Immune aging is thus both a failure of defense and a failure of regulation. The system loses precision, loses balance. Can immune function be restored? Partially, possibly. Vaccination remains important, even if responses are weaker. Higher dose or adjuvanted vaccines are used in older adults to try to compensate for immunosenescence. Clearing senescent cells, which reduces SASP, can improve immune function. In mice, Senolytics restore some aspects of immune response. NAD+ boosting has shown some promise in animal models of improving immune function, possibly by enhancing mitochondrial health in immune cells. Exercise is one of the most robust interventions. It improves immune surveillance, reduces inflammaging, enhances vaccine responses. Adequate sleep is critical. Sleep deprivation impairs immune function. Chronic poor sleep accelerates immune aging. Nutrition matters. Malnutrition, vitamin deficiencies, particularly vitamin D, zinc, selenium, all impair immune function and are more common in older adults. There's also interest in thymic regeneration, finding ways to restore thymic function and boost T cell production. Some experimental approaches, growth factors, gene therapies are being explored, but none are clinically ready. And broadly, interventions that reduce the other hallmarks, improving proteostasis, mitochondrial function, clearing senescence, supporting the microbiome, all may indirectly improve immune aging by reducing the chronic inflammatory burden and supporting immune cell health. The immune system is not separate from the rest of aging biology. It's embedded in it. Its decline both causes and results from the accumulation of damage in other systems. And so we come to the end of the 12 hallmarks, 12 windows into the biology of aging, each one a thread, together a tapestry, not a simple picture, but a complex interconnected whole. Aging is not one thing breaking. It's a network of systems gradually losing coordination, accumulating damage faster than repair can keep up, drifting from the tightly regulated state of youth toward the dysregulated state of old age. But understanding this, mapping it, naming the parts brings clarity, and clarity brings a kind of peace. It's not mysterious, it's not punishment, it's biology, and biology can be understood. Rest now. Breathe. We're going to step back and look at the connections, the way the hallmarks interact, before moving on to what can be done. If anything, the 12 hallmarks are not independent. They're not like separate diseases that happen to occur at the same time. They're deeply interconnected, each one influencing the others, creating feedback loops that can amplify decline. Think of it as a network. Each hallmark is a node. Lines connect them, representing causal relationships. Damage to one node sends ripples through the network, affecting others, which feed back and worsen the original problem. Let's trace a few examples. Genomic instability drives multiple other hallmarks. DNA damage can alter the expression of genes involved in epigenetic regulation, causing epigenetic alterations. It can damage genes encoding proteostasis machinery, impairing protein quality control. It can damage mitochondrial DNA, causing mitochondrial dysfunction. It can activate senescence programs, increasing cellular senescence. And it can impair stem cell function, contributing to stem cell exhaustion. Mitochondrial dysfunction creates a vicious cycle. Damaged mitochondria produce more reactive oxygen species, which cause more DNA damage. More protein oxidation, more lipid peroxidation. ROS also damage the mitochondria themselves, worsening their dysfunction. And mitochondrial dysfunction impairs autophagy because autophagy is an energy-demanding process. So damaged mitochondria accumulate because the system that should clear them is itself impaired. Cellular senescence amplifies inflammation. Senescent cells secrete SASP factors which spread inflammation. Chronic inflammation damages neighboring cells, causing more DNA damage. More mitochondrial dysfunction, more proteostasis failure, which can trigger more senescence. And inflammation disrupts the stem cell niche, impairing stem cell function. It also promotes dysbiosis, altering the gut microbiome, which further increases systemic inflammation. Epigenetic alterations affect almost everything. Changes in DNA methylation and histone modifications can downregulate autophagy genes, DNA repair genes, antioxidant genes, anti-inflammatory genes. This worsens proteostasis, genomic instability, mitochondrial dysfunction, and inflammation. Epigenetic drift is thus both a consequence of damage and a driver of further decline. Deregulated nutrient sensing locks cells into growth mode when maintenance is needed. Chronic mTOR activation inhibits autophagy, allowing damaged components to accumulate. It drives protein synthesis even when proteostasis is failing, overwhelming the system. It promotes senescence in some contexts, and it increases oxidative stress, feeding mitochondrial dysfunction and genomic instability. Stem cell exhaustion limits the body's ability to compensate for other hallmarks. When tissues lose regenerative capacity, damage accumulates. Senescent cells aren't replaced. Dysfunctional cells persist. Inflammation isn't resolved because the new cells that should replace damaged ones aren't being produced. Dysbiosis contributes to systemic inflammation, which affects every other hallmark. Inflammaging worsens mitochondrial function, drives senescence, impairs autophagy, damages DNA, alters epigenetics, exhausts stem cells, dysregulates nutrient sensing, and further impairs immune function. You can see how this works. One problem creates another. The second problem worsens the first. Feedback loops form. The system becomes locked in a state of accelerating decline. This is why aging is so hard to reverse once it's advanced. It's not just one thing to fix. It's a deeply entangled network, where every intervention has to contend with the fact that the system is fighting itself. But this also suggests a strategy. If you can intervene at key nodes, particularly upstream ones, you might be able to slow or partially reverse multiple hallmarks at once. For example, enhancing autophagy could clear damaged mitochondria, reduce protein aggregates, remove senescent cells, all at once. Clearing senescent cells could reduce inflammation, improve the stem cell niche, restore tissue function, and reduce further senescence. Improving nutrient sensing could enhance autophagy, reduce oxidative stress, support proteostasis, and improve metabolic health. This is the logic behind many proposed interventions. Target a key node, and the effects ripple through the network. But it's also why simple interventions, one drug, one supplement, are unlikely to be magic bullets. Aging is a system-level problem. It requires system-level solutions, or at least multi-targeted approaches. And it's why the Hallmarks framework is so valuable. It gives researchers a common language, a shared map. When someone proposes an intervention, the questions are, which hallmarks does it affect? How many? Are they upstream or downstream? What are the trade-offs? For you, lying here, the takeaway is this. Aging is complex but not incomprehensible. It's a network of interconnected processes, each understandable, each measurable. And while that network is difficult to untangle, understanding it is the first step toward influencing it. No single action will stop aging, but many small actions across diet, exercise, sleep, stress, environment might shift the balance, slow the accumulation, support the maintenance systems, delay the onset of dysfunction, or maybe not. Maybe aging is too deeply woven into biology to be significantly altered by lifestyle. That's still an open question. But understanding the system is valuable regardless because it removes mystery, replaces fear with knowledge, and allows for informed choices. Let it settle. The hallmarks, the connections, the feedback loops, a living network playing out in your body right now as you rest, breathe. We're moving now to how aging is measured, how scientists try to capture biological age in a number, a marker, something trackable.biomarkers of aging. Let's continue. Chronological age is easy to measure. You count the years, but biological age, the actual state of your body's systems, is much harder to quantify. And yet, measuring biological age is essential if we want to test whether interventions actually slow aging. You can't run a lifespan study in humans. It would take 80 years. But if you had a reliable biomarker of aging, something that changes predictably with biological age and predicts future health and mortality, you could measure it before and after an intervention, see if it improves, and infer that the intervention is working. This is the promise of aging biomarkers, and there's been intense research to develop them. What makes a good aging biomarker? Several criteria. It should change with age, consistently, across individuals. It should predict health outcomes, mortality, disease risk, better than chronological age alone. Ideally, it should be causally related to aging, not just correlated. It should be measurable relatively easily, ideally from blood or other accessible samples. And crucially, it should respond to interventions that are believed to slow aging. Many candidates have been proposed. Let's look at a few. Telomere length was one of the first widely studied biomarkers. As discussed, telomeres shorten with age. Shorter telomeres are associated with higher mortality and more age-related disease. But the relationship is noisy. Telomere length varies widely between individuals and even between cells in the same individual. It's affected by many factors beyond aging, genetics, stress, lifestyle, and interventions that extend lifespan in animals don't always affect telomere length, suggesting it's not a perfect marker. Epigenetic clocks are currently the most promising biomarkers. These are algorithms trained on large datasets that predict age from DNA methylation patterns. The first clock, the Horvath clock, was published in 2013 and used methylation at 353 specific sites in the genome to predict age with remarkable accuracy. Since then, many other clocks have been developed. Some are trained to predict chronological age. Others are trained to predict health outcomes like mortality or disease risk. Often called aging clocks or mortality clocks, Phenoage and Grimmage are examples, and they often outperform chronological age in predicting who will die or get sick in the next decade. Epigenetic clocks are exciting because they're based on a biological process. Epigenetic alterations, that's a hallmark of aging, and they seem to respond to interventions. Caloric restriction, exercise, certain drugs, all have been shown to affect epigenetic age, though the effects are often modest, but there are caveats. Epigenetic clocks are still correlational. We don't fully understand why these specific methylation sites change with age, or how they causally contribute to aging, and different clocks sometimes give different answers. Your epigenetic age might be younger than your chronological age on one clock, older on another. Inflammatory markers like CRP, IL-6, TNF-alpha are elevated with age and associated with poor health outcomes. They're easy to measure from blood, but they're highly variable, affected by acute illness, diet, stress, and their downstream consequences of many processes, not specific to aging per se. Metabolic markers like blood glucose, lipid profiles, insulin sensitivity change with age and predict disease. But again, they're influenced by many factors and are more markers of metabolic health than aging per se. Frailty indices are clinical tools that assess physical and functional decline. They consider things like walking speed, grip strength, cognitive function, activities of daily living. High frailty predicts mortality and disability. But frailty is the outcome of aging, not a molecular marker of the underlying process. Composite panels combining multiple markers, blood proteins, metabolites, clinical measurements, epigenetic clocks into a single score are being developed. The idea is that no single marker captures aging fully, but a combination might. Companies are now offering commercial aging tests, usually based on epigenetic clocks, where you send in a saliva or blood sample and get back your biological age. These are interesting, but they should be interpreted cautiously. The clocks are good predictors at the population level, but individual predictions have uncertainty. And it's not yet clear whether tracking your epigenetic age over time and trying to reduce it actually translates into better health or longer life. The field is moving fast. New biomarkers are being discovered. Machine learning is being applied to find complex patterns in large datasets that predict aging. But we're still in the early stages. For now, the best biomarkers are probably composite ones, looking at multiple systems, physical function, metabolic health, immune markers, epigenetic age. Together, no single number will capture the complexity of aging. And for individuals, the most reliable markers are still simple ones. How you feel, how you function, whether you can do the things you want to do, whether you're developing age-related diseases. These may not be molecular, but they're real. The science of aging biomarkers is advancing, and it's exciting. It offers the possibility of tracking aging in real time, testing interventions, personalizing approaches. But it's not magic. A number on a test is just a number. What matters is health, function, quality of life. Don't be seduced by the promise of a perfect aging clock, and don't be discouraged if one tells you you're older than you hoped. These are tools, imperfect, evolving. Use them with curiosity, not fear. Breathe. Rest. We're moving now to the question you've probably been waiting for, the question everyone asks. Can we slow aging? What actually works? Let's explore. The science of aging has exploded in recent decades. We understand more about the mechanisms than ever before. And with that understanding comes the tantalizing question, can we intervene? Can we slow, stop, or even reverse aging? The answer is maybe, partially, in some ways we're starting to. But there's a vast gap between what's been shown in laboratory animals and what's been proven in humans. And the space between is filled with hype, oversimplification, and commercial exploitation. So, let's be careful. Let's look at what the evidence actually supports. Lifestyle interventions are the most robust, accessible, and proven approaches. They won't make you immortal, but they can shift the trajectory, compressing the period of disability and disease into a shorter window at the end of life. A concept called health span extension. Caloric restriction is the gold standard in aging research. In worms, flies, mice, rats, and many other species, reducing calorie intake without malnutrition extends lifespan, sometimes dramatically. A 30 to 40% reduction in calories can extend mouse lifespan by 20 to 40%. In primates, the results are more mixed. 2 long-term studies in rhesus monkeys gave somewhat different results, but both showed health benefits, less cancer, less cardiovascular disease, better metabolic health. Lifespan extension was modest, if present at all, but health span, the period of life in good health, was clearly improved. In humans, we don't have lifespan data, because it would take too long to collect. But the calorie trials, comprehensive assessment of long-term effects of reducing intake of energy, tested moderate caloric restriction in healthy, non-obese adults. Participants reduced calories by about 12% on average over 2 years. The results were positive. Weight loss, obviously, but also improvements in metabolic markers, reduced inflammation, lower blood pressure, better cholesterol, improved insulin sensitivity. Epigenetic age measured by certain clocks slowed. Quality of life and mood improved. But adherence was hard. Many participants couldn't maintain the restriction, and 12% is mild compared to the 30 to 40% used in animal studies. So caloric restriction may work in humans, but it's difficult, and we don't know if it actually extends lifespan. For most people, long-term severe restriction is not realistic or desirable. Intermittent fasting and time-restricted eating are alternative approaches that may capture some of the benefits of caloric restriction without continuous deprivation, by limiting eating to a specific window each day, or by fasting for full days periodically. You extend the time the body spends in a fasted, maintenance-oriented metabolic state. Animal studies show benefits. Human studies are emerging, showing improvements in weight, metabolic markers, inflammation. But long-term, large-scale trials are lacking. And like caloric restriction, adherence can be challenging. Exercise is perhaps the single most powerful, evidence-based intervention for healthy aging. It affects almost every hallmark. It improves mitochondrial function, stimulates autophagy, reduces senescent cell burden, enhances proteostasis, supports stem cell function, reduces inflammation, improves immune function, maintains muscle and bone, supports cognitive health, improves mood. Both aerobic exercise and resistance training are important. Aerobic exercise improves cardiovascular and metabolic health. Resistance training maintains muscle mass and strength, counteracting sarcopenia. The evidence is overwhelming. People who exercise regularly live longer, have less disability, fewer chronic diseases, better cognitive function. The dose response is strong. More exercise, up to a point, means more benefit. And unlike caloric restriction or fasting, exercise is something most people can do, can enjoy, can sustain. Sleep is critical. Sleep deprivation accelerates aging markers, impairs immune function, worsens metabolic health, increases inflammation, impairs cognitive function, and shortens lifespan in animal models. Good sleep, 7 to 9 hours for most adults with consistent timing and good quality, supports almost every aspect of health. It's when the brain clears metabolic waste, when tissues repair, when immune surveillance is heightened. Diet quality matters beyond just calories. Diets rich in plants, whole grains, healthy fats, fish, low in processed foods, refined sugars, and excessive red meat are consistently associated with longer, healthier lives. The Mediterranean diet is one example, supported by strong evidence, specific nutrients and compounds, polyphenols, omega-3 fatty acids, fiber, vitamins, minerals all contribute. But supplements, isolated vitamins or antioxidants, have generally not shown the benefits that whole foods do. The synergy of nutrients in whole foods seems to matter. Social connection and purpose are less tangible but equally important. Loneliness and social isolation are as harmful to health as smoking. Strong social ties, meaningful relationships, a sense of purpose, all are associated with longer, healthier lives. Stress management matters. Chronic stress elevates cortisol, impairs immune function, accelerates telomere shortening, promotes inflammation. Practices like meditation, mindfulness, yoga, anything that reduces chronic stress, may have protective effects. These lifestyle factors are not magic. They won't stop aging, but they can slow it, shift the trajectory, compress morbidity, add years of healthy life. And the evidence for them is strong. Much stronger than for any drug or supplement currently available. So, if the question is, can we slow aging? The answer, supported by evidence, is yes, somewhat, through how we live. But people want more. They want pills. They want therapies. They want technological solutions. And that brings us to the next section. Beyond lifestyle. There's a growing field of pharmacological and biotechnological interventions aimed at targeting the hallmarks of aging directly. Some are in early human trials. Some are still purely experimental. None are proven to extend human lifespan. But they represent the frontier of aging research, where the science is moving from understanding to intervention. Let's look at a few. Senolytics are drugs that selectively kill senescent cells. As discussed, senescent cells accumulate with age and secrete inflammatory SOS factors. Clearing them in mice extends lifespan and health span dramatically. The first widely studied senolytic combination is dasatinib plus quercetin, DQ. Dasatinib is a cancer drug. Quercetin is a flavonoid found in plants. Together, they seem to induce apoptosis in senescent cells, though the exact mechanism is not fully understood. In mice, DQ treatment clears senescent cells from tissues, reduces inflammation, improves physical function, and extends lifespan, especially when started in old age. In humans, early trials are underway. A small trial in patients with idiopathic pulmonary fibrosis, a lung disease where senescence is thought to play a role. SHOWED that D plus Q improved physical function and reduced markers of senescence. Other small studies in older adults have shown reductions in inflammatory markers, improved vascular function, and modest improvements in physical performance. But these are very early results. The trials are small, short, often open-label without placebo controls. Long-term safety is unknown, and it's not clear how often treatment would need to be repeated, or whether it would work in healthy aging, not just disease. Other senolytics are being developed. Ficitin, another flavonoid. Navitoclax, a cancer drug. UBX0101, a small molecule tested for osteoarthritis. Some have shown promise in animal models. Human data is sparse. The field is exciting, but it's important not to overhype. Senolytics are experimental. They're not available for anti-aging use outside of clinical trials. Self-experimenting with dasatinib, a chemotherapy drug, or even high-dose quercetin without medical supervision is risky. Metformin is a diabetes drug used for decades, with a strong safety profile. It lowers blood sugar by improving insulin sensitivity and reducing glucose production in the liver. But metformin may have effects beyond glucose control. Epidemiological studies suggest that diabetics on metformin have lower cancer rates and, in some analyses, lower mortality than diabetics on other medications, and even than non-diabetics in some comparisons. Though this last point is debated. METFORMIN activates AMPK, which promotes autophagy, improves mitochondrial function, and shifts metabolism toward maintenance. It may also reduce inflammation and improve gut microbiome composition. Animal studies show lifespan extension in some contexts, though results are mixed depending on dose, species, and genetic background. The TAME trial, targeting aging with metformin, is designed to test whether metformin can delay the onset of age-related diseases. In non-diabetic older adults, it's a landmark study, the first large trial designed to test an intervention specifically for aging, not a specific disease. But TAME hasn't started enrollment yet, despite being proposed years ago. Funding and regulatory challenges have delayed it, and even when it runs, it will measure disease outcomes, not lifespan, because that's more feasible. Some doctors prescribe metformin off-label for potential anti-aging benefits, especially for people with prediabetes or metabolic syndrome. But for healthy individuals, it's not standard and the evidence is not conclusive. Rapamycin, which inhibits mTOR, is one of the most robust lifespan-extending drugs in animal models. Mice treated with rapamycin live longer, have less cancer, better immune function, improved cognition. But rapamycin is an immunosuppressant. It's used to prevent organ rejection after transplants. Long-term use increases infection risk, can cause metabolic side effects, and in high doses has significant toxicity. Some researchers and longevity enthusiasts take low dose, intermittent rapamycin off-label, hoping to capture the benefits without the severe immunosuppression. Small studies are beginning to test this in humans, but the data is very preliminary. It's possible that a safer rapamycin analog, a rapalog, could be developed that has the longevity benefits without the immunosuppressive side effects. Research is ongoing. NAD boosters, like NMN, nicotinamide mononucleotide, and NR, nicotinamide riboside, are supplements that aim to raise levels of NAD, a molecule involved in cellular energy metabolism that declines with age. NAD+ is required for sirtuin activity and for mitochondrial function. Raising NAD+ in mice improves mitochondrial health, enhances metabolism, and in some studies, extends lifespan or improves HEALTHSPAN. In humans, NMN and NR are available as supplements. Some small studies showed they raise blood NAD+ levels and improve some metabolic markers, but the effects are modest and the long-term benefits are unproven. There's significant commercial hype around NAD+ boosters. They're sold as anti-aging supplements, often with exaggerated claims. The science is interesting but early. They're probably safe, but whether they meaningfully slow aging in humans is unknown. Partial reprogramming is one of the most exciting and speculative frontiers. It's based on the discovery that for transcription factors, called Yamanaka factors, can reprogram adult cells back to a pluripotent, embryonic-like state. This is how cloning works. But full reprogramming erases cell identity, which is dangerous in a living organism. It could cause tumors or loss of tissue function. Partial reprogramming. Brief exposure to these factors can reset some aging markers, epigenetic patterns in particular, without fully reverting cells to pluripotency. In mice, partial reprogramming has been shown to rejuvenate tissues, improve vision in aged eyes, extend lifespan after injury, and in some models, extend overall lifespan. The idea is that you could reset the epigenetic clock, restoring youthful gene expression patterns, without losing cell identity. But this is extremely early. It's not a therapy. It's not even close to clinical trials in humans for aging. The risks are enormous, particularly cancer, if the reprogramming is not precisely controlled. Companies like Altos Labs and Calico are investing heavily in this area, but it will be many years, maybe decades, before we know if it can be done safely and effectively in humans. Summary of the frontier: there are promising leads: Senolytics, metformin, rapamycin, NAD boosters, partial reprogramming, and others. But none are proven in humans for slowing aging. The evidence is mostly from animals or small, early-stage human studies. Long-term safety and efficacy are unknown. The field is moving from basic science toward translation, but slowly, carefully as it should be. Aging is complex. Interventions will have trade-offs, and human lifespan is long, making definitive trials difficult. So be excited, but skeptical. Be curious, but patient. The science is real, but the hype often outruns the evidence. And for now, the most reliable interventions remain lifestyle, diet, exercise, sleep, stress, social connection. Boring, perhaps, compared to high-tech therapies, but evidence-based, accessible, safe. Let that settle. The frontier is far away. The basics are right here. Rest. Breathe. We're coming to the close now, integrating everything, bringing it back to you, to this moment. You've traveled far tonight, through the inner landscapes of your cells, through hallmarks and mechanisms, feedback loops and interventions. You've heard about DNA damage and telomeres, mitochondria and senescence, inflammation and microbes. It's been a lot. And you didn't need to remember it all. You just needed to be here, listening, letting it wash over you. So what does it all mean, really? What should you take from this as you lie here on the edge of sleep? First, this aging is not your fault. It's not a failure of willpower or discipline or virtue. It's biology. It's physics. It's the consequence of being a complex, organized system in a universe that tends toward entropy. You are not broken because you're aging. You're human. Second, aging is not one thing. It's 12 things or more, all interconnected. There's no single cause to blame, no single fix to seek. It's a system, and systems require balance, multiple inputs, modest, sustained efforts across many dimensions. Third, you have some influence, not control, influence. How you eat, how you move, how you sleep, how you manage stress, how you connect with others. All of these shape the trajectory. Not dramatically, perhaps. Not enough to guarantee anything, but enough to matter statistically, measurably in the long arc of a life. Fourth, science is learning. We're not helpless. The map of aging is clearer than it's ever been. Interventions are being tested. Some will fail. Some will work. Progress is real, even if it's slow. And fifth, most importantly, you are more than your biology. Yes, you are cells and molecules and processes. Yes, aging will happen, but you are also experience, consciousness, relationships, meaning. The quality of your life is not reducible to the length of your telomeres or the state of your mitochondria. It's about what you do, what you feel, what you create, who you love. Understanding aging can free you from fear, not because you'll escape it, but because you'll see it clearly as process, not as failure. And seeing it clearly, you can focus on what actually matters, living well. Whatever that means to you. Tonight, You've given yourself the gift of understanding. You've learned about the slow transformation happening inside you, the drift from youth to age, the dance of damage and repair. And in understanding, there's a strange sort of peace, a sense of being part of something much larger, a sense of acceptance. You are a temporary pattern, a brief arrangement of matter into something beautiful and complex. And aging is the slow return towards simplicity. It's not good or bad, it just is. And you are here, now in the midst of it, breathing, thinking, feeling. That's enough. That's more than enough. That's everything. Let your body rest. Let your mind quiet. You've done the work of learning. Now let sleep take you, gently, softly, into the dark. Thank you. Truly, deeply, thank you for being here tonight, for choosing to spend these hours with me exploring the science of aging, drifting through the microscopic cosmos within your own body. I'm honored to have been your companion on this journey. If this episode has helped you, if it's given you peace or understanding, or simply a gentle place to rest, I'd be grateful if you'd share that. A like, a subscription, a comment, or share it with someone else who might find comfort in falling asleep to science. These small gestures help this work continue, help it reach others who need a calm, curious voice in the night. And if you wake later, if you find yourself in the deep quiet hours and need a familiar voice to guide you back towards sleep, this episode will still be here. You can return to it. Let it play in the background. Let the words wash over you again, familiar, soothing, a river carrying you back toward rest. Remember, aging is not something to fear. It's something to understand. And understanding brings peace. You are part of the great flow of life, of time, of change. You're not separate from it. You're in it, of it, made of it. And that's a beautiful thing. So now, as we close, let's return to the simplest thing. The breath. The gentle rise and fall of your chest. The quiet rhythm that's been with you since your first moment and will be with you until your last. Feel the breath. Cool air in, warm air out, in and out. Nothing to do. Nowhere to be. Just this. Just breathing. Just being. The universe is vast. Time is long. But this moment, right here, right now, is yours. And in this moment, you are safe. You are resting. You are exactly where you need to be. Let the darkness hold you. Let the silence surround you. Let sleep, whenever it comes, come gently. And know that the same processes we've explored tonight, the repair, the recycling, the renewal, are happening even now, even as you rest. Your body is caring for itself. Your cells are doing their work. You don't have to do anything but be here. Breathe, rest, sleep, and drift into the cosmic atlas of your own being, a galaxy of cells, a universe within, vast and ancient and impossibly, beautifully alive. Good night, sleep well, and thank you again for being here.